promote the research by faculty, residents, students, PhD scholars at JIPMER. And this day is the past year. It is the staff of JIPMER is informed about this scientific research. So today is the culmination of the 4 weeks session that is research work is being last year. Medical category by NIRF, National Institute Ranking Framework 2025. JIPMER is known for its medical education and special patient care as well as biomedical research. 1,000 residents and 30 PhD students across various departments who are engaged in the research. The vision of the division of research, I say a wing of research at JIPMER, is to enhance the fundamental, clinical, and translational research. This wing biology and advanced data analytics. We have Central Animal House at JIPMER, which is state-of-art animal research facility that has been designed to support cutting-edge biomedical research and training in the institute. The research infrastructure is always updated and enhanced every year with latest software and project management support. Faculty from JIPMER has received several grants at the national and international levels from ICMR, DBT, DHR, to many international collaborations as PI and co-PI. 200 research projects are there going on with extramural funding at JIPMER, with total sanctioned amount of 107 crores funded by different agencies. The researchers from JIPMER contributed to 1,050 scientific publications in just the year of 2024. As a part of its commitment to innovation and development, JIPMER filed 30 patent applications and 9 were granted. The rest are under consideration. Recently, we had 4 part of research day activity, focusing on capacity. Digital droplet PCR, health technology assessment, and introduction the culmination of the research day. And we have host of exciting research related award and prizes. And Dr. Venkatasubramanian will be sharing his thoughts on making a clinical scientist, the obstacles and the path ahead. We all are very sure that he's going to inspire us all present over here. At last, I wish to conclude that we belong to the medical fraternity. It's a noble profession to serve the suffering mankind. Human being. I have no doubt that this Research Day celebration, the research training at JIPMER is going to, this research training all through. I pray you all realize your inspirations, working with commitment and passion, sincerity and dedication. Thank you. Jai Hind.
Thank you, ma'am.
Now, we request the dignitaries to light the lamp that symbolizes truth, knowledge, and understanding. Dr. Venkatasubramanian Ganesan to deliver the keynote address.
Good morning and happy Women's Day to all. Respected Director, Professor Bhavana Pade Madam, the Dean, Professor Thapa, and then I understand Madam Professor Sarkar is the one who manages this stage. And of course, there are several dignitaries of the dais. Respected teachers inspire me the most. Good to see so many students. I understand most of you are in the initial years of your be here. It's a wonderful institute, an institute I always hold in very high regard. And I was really overwhelmed when I got an invite from your esteemed institute, madam. Thank you. So the title of the making of a clinician scientist. I have given a tagline: the obstacle is the path. Many of this saying what we see as an obstacle in various spheres of life may indeed be the path. So I work at the National Institute of Mental Health and Neurosciences, Bengaluru. For some of you that cannot have visited the institute, it has a beautiful campus. What we see here is the department in which we work. And I invite you all, when you get an opportunity to visit Bengaluru, please come to our institute. The prologue of my talk introduce a character which I will keep revisiting throughout my interaction. So I introduce this character who is Dr. Pangloss. Some of you may be aware of this character, Dr. Pangloss, who is the eternal optimist, who finds this world to be the best of all possible worlds, with their unique training in medicine and in scientific endeavor can enhance patient care, improve medical education, and increase the prosperity of biomedical enterprise. This is a paper by Archer where it is summarized that if you look at from 1901 and look at the Nobel laureates, especially in medicine and physiology, 101 out of 72 were actually people that were in medical. These are essentially clinicians. Now, if we look at clinician scientists, it need not always be restricted to lab-based work. There are other facets enterprise, which may be a research-focused position. These are physicians who spend a substantial proportion of their time in research outside of their clinical practice, but not in the lab. There are also research trend in terms of the proportion of medically trained students that take this career path is slowly decreasing. So, which has kind of raised this question, why this breed of people are reducing. The number of young students embarking on a path to become physician scientists has been kind of declining. So in that context, there are multiple levels of challenges. Obstacles could be looked at. Look at clinicians versus scientists. They may be two different species. This is in a lighter vein has been shown in this kind of diagram where by training, philosophy, motivation, and research style, the doctors and scientists might well be in two different phyla. And if you look at the thinking pattern and the attributes of clinical scientists, clinicians are motivated by the need for immediate action. Many a time it has to save someone's life. But scientists are conditioned to avoid rushing to judgment. With a belief, and that's what will push the envelope and push the understanding and knowledge. For clinicians, errors are potentially more threats, whereas for scientists, errors are inevitable manifestations of the kind of creative process they are engaged in. So we see that in terms of the paradigms, the attributes, and the kind of orientation they have to their respective fields look antithetical. We see them quite opposite in this case. So this is at one level. So if you look at the attributes, they seem to be quite divergent. In addition, if you look at the recent advances in the research, there has been a big gap which is increasing between the clinical practice versus basic biomedical research. The advances in either have become so much that interaction between these two lines of fields is becoming more and more challenging. And over the past decades, there is also a lot of sophistication that has been happening. I'm happy to learn that there are quite a bit of facilities, madam, you listed, right from the animal model research to a lot of omics-based research, then advanced statistical software, then clinic-based research. Each of these fields are expanding, and so they are becoming more and more like a silos, so that interaction between them and understanding what is happening in another subdomain is becoming more challenging. So that discourages clinicians to get out of their comfort zone and try to learn more about what is happening in not so closely related field of clinical medicine. If you look at the international scenario, uh, so there one has to go through what is called as an MD PhD program to be seen as a qualified clinician scientist, which is invariably a long duration program, and it is a less lucrative career option. And if you look at the way in which science is progressing, because of the technological advances, at one level, technology has enabled so much. At the other level, the deductive reasoning, which was the cornerstone of the clinical approach and intuition, is slowly declining. And it is coming in a good way, more and more evidence-based research, more and more technology-driven research rather than theory-driven research. Whereas with respect to an Indian scenario, of course, the major obstacle is the patient load. The kind of patient population numbers we handle is humongous and the challenge is phenomenal. And we do not have protected time, and we have limited mentors that will help us to embark in the path of clinician scientist career, and also certain important cornerstones of research, namely understanding the principles of design, and also the statistical techniques look so much overwhelming. And also getting grants is becoming more and more difficult. So because of all this, anybody that finishes MBBS or MD thinks twice whether I need really to kind of worry about getting into all these uncharted, less lucrative territory. So that's another major challenge. Now, if we ask Dr. Pangloss, okay, these are the obstacles. Dr. Pangloss, what do you think? So if I have to take that kind of perspective, perhaps these obstacles are the path which will help us to adapt to the changing scenario and equip ourselves adequately to become effective clinician scientists. For instance, while patient load is a major challenge, in certain setups, getting patients for research itself is a major challenge. So compared to that, having a good patient load is a boon. It would be helpful, at least in those kinds of centers, to primarily look at patient-oriented translational research. And if the clinical data that is getting collected, now that more and more digital technology is implemented in most of the institutes, a carefully thought through clinical data collection will be very helpful to do high quality research amidst heavy patient load. Then comes the lack of protected time, which of course compels us to be very efficient in time management. And it is impossible for an individual to get excellent expertise in different fields. Now, given this challenge, now the constraint is to collaborate, to look at people from other fields and have close dialogues. And also, very importantly, we need to evolve programs which will mentor young researchers from diverse fields. I'll be just sharing some of the research experiences and learnings that I've had. I would say that several active and young and dynamic students that came from different fields are the ones who contributed to the ongoing research program. Coming to the challenge of difficult to secure grants, there is this concept of N of 1 trials. In the sense, we can actually do a systematic research experiment using just one challenging patient, of course, following all the ethical and related research design requirements, which can be actually considered as a personalized medicine and will go a great way in enhancing the patient care. So for such kind of research, not much of resources is required. All that is needed is a curious mind, time, and applied clinical evaluation and careful monitoring. Of course, that has to be guided by a passion and some kind of innovative out-of-thinking box to try different newer interventions. And also there is this question of small science versus big science, where the big science needs lots and lots of funding, whereas these kinds of N of 1 trial could be small science, but such small science are the ones that have led to interesting medical breakthrough. For instance, the discovery of H. pylori or the discovery of several interventions, including, say, intravascular catheterization and so on and so forth, have come from certain initial simple observations perfectly followed upon. And such research attempts over the course of time, as we gather more and more, publication will help us to secure more grants, especially if we get a right set of collaborators. And there has to be system level changes. The initial points I mentioned were more like an individual level changes. It has to be complemented with system level changes, which in my view, the mentorship programs are amounts. And I'm very delighted to see how much research emphasis is being given on the UG curriculum. I think the UG students are amongst the extremely gifted one. You have so much of opportunities, and this is a time at which one has to hone the fundamentals of research. And of course, there has to be certain major administrative changes in terms of departments having a mix of both clinicians and basic science. Next, about 5-10 minutes, I will just dwell upon some of my learnings from the schizophrenia related research that I have been involved in. Now, all of us will be familiar with this challenging disorder, schizophrenia. And many of us will be familiar with this Nobel laureate I have put forth in this slide, who's Professor John Nash. Professor John Nash lived with schizophrenia. And at one point in time, he was asked by a fellow mathematician, Nash, how could you believe that recruited by aliens from outer space to save the world? For which Nash reply was, because the ideas I had about supernatural beings came to me in the same way that my mathematical ideas did. This statement profoundly reflects the essence of the disorder, essentially characterized by hallucinations and delusion, where the reality perception when someone has the symptoms of schizophrenia. If you look at some of the studies that are published, especially, and the one which was published in Lancet many years before, which looked at among the entirety of medical disease and disorder, perhaps it's the most disabling, especially if you look at the acute stage of symptom severity. So to handle this challenging disorder, we run this specialized clinic program, which is called as Individualized Schizophrenia Treatment and Reintegration, in which we have an inclusive approach that has a medication and other biology-based intervention, which is complemented by psychological and social and mind-body medicine-based approaches. Now, this slide, I thought I will put the first case report I published 25 years before when I was an MD resident. So before joining MD Psychiatry at NIMHANS, I was doing MD General Medicine in Stanley Medical College, where I finished my MBBS. I had some kind of curiosity towards understanding more about psychiatric disorders, which became more like a passion after I finished the UG posting. We had about 2 weeks of posting in psychiatry. So during my MD General Medicine, initially I was again posted in psychiatry wards in the peripheral. For about a month I spent, my curiosity became much, much more. But I had equally compelling passion for medicine as well. But I thought I will choose the psychiatry. I don't know the road that I had not taken, but I'm happy that I have chosen psychiatry. So once I entered into my MD training, the medicine interface was always there in the back of my mind. So something which intrigued me was the kind of side effects with antipsychotics. One is, of course, we know that it causes Parkinsonism-like side effects, which are called as extrapyramidal symptoms. So this was the first case report which I published with my mentor, Professor Gangadhar, who is also the chair of the National Medical Committee now. So my interest in the clinic and research was having a lot of medicine interface to begin with. What intrigued me was the metabolic side effects. Almost all antipsychotics cause quite a bit of metabolic side effects, specifically obesity. So I used to wonder, why should a side effect happen so consistently with majority of antipsychotics that we treat? So that led me to do some research under the guidance of my mentor, where I embarked on some findings related to the insulin abnormalities. To begin with, it was a clinical research. Later, we collaborated with the neurochemistry department, which led on to some more findings related to this insulin-like growth factor, which essentially emphasized the role for neuroplasticity in this disorder. And I had a good opportunity to be involved in several brain imaging research. The purpose of those research studies were to connect the biology, the systemic observation with brain changes. So what we see here is a functional MRI research, which I was part of when I became a faculty. The student that is shown here, now he is a doctor who's basically coming from a medical biotechnology background, Dr. Sunil Kalmadi. Now he's working as a professor in Canada with a data science approach. And he's the one who did this study showing how brain abnormalities in schizophrenia can be understood using functional MRI technique. Now, based on several of these studies, a simplistic understanding that we arrived at is in patients with schizophrenia, certain parts of the brain could be hyperactive. Say, for instance, the temporoparietal junction, and certain parts of the brain could be hypoactive. Say, for instance, the prefrontal cortex. The hyperactive regions perhaps may be generating the hallucinations and delusions, whereas the hypoactive regions may be contributing to amotivation and related symptoms, which are called as negative symptoms. So this is one line of simplistic conclusion we could come across with several of the brain imaging studies. And when we combine that with another parallel line of research, looking at the neurotrophic factors and neuroplasticity related aberrations, we came up with a model where the neuroplasticity and neuroimmunological interactions may be driving the pathophysiological basis of schizophrenia. So this set of research studies were possible thanks to my mentors. Apart from Professor Gangadhar, there is Professor Keshavan, who's currently in the Harvard, and Professor Ravi, who used to head the neurovirology department at NIMHANS. And I also had an excellent collaborator, Dr. Manojit Devnath, who helped us to do these studies. Essentially, the model emphasizes the role for neuroplasticity abnormalities. Now, some of you who are into the middle part of the medical training, especially the ones who have completed the physiology, will be familiar with the concept of neural plasticity. That is the brain's ability to change its architecture based on the experience. Now, the plasticity is interesting because we have a window where we can use some interventions to modulate the brain. So in that context, we were looking for any simplistic intervention that can modulate the brain plasticity through which we can treat these symptoms. So we came across this principle: weak direct current can modulate neuroplasticity. So there is a neuromodulation technique that is called as transcranial direct current stimulation, which uses battery delivered current in the strength of 1 to 2 milliampere, which is absolutely safe and very well tolerated. And that was reported to show some improvement in symptoms of schizophrenia. Enthusiasm, from the brain imaging and the biology research made us to import this device. So when the device was imported, none of us knew how to use it. So there was the technical person from the company who helped us to set it up. So that was our anniversary day. So I told my wife, today we are getting this new electrical stimulation device. Maybe as a pilot, kind of got it here, and none of us knew how it is going to feel like. There are studies that show that it can be safely applied in healthy people for a investigative purpose as a single session. So I'm going to try that today. So she told, today is our anniversary day. By the time you come back, you make sure you remember me. Right. So it happened about a decade before, more than a decade before. So that's how this TDCS research started. This is the research intervention protocol that we follow. 5 days of treatment in people that have persisting auditory hallucination despite antipsychotic medication. So when we use this, we found very impressive results. These are the set of students that come from different backgrounds. Anushri comes from the psychology background. Dr. Shiv Kumar is actually a clinician scientist who finished his MBBS and PhD. And others are colleagues, both Janardhan and Mahavir are colleagues from psychiatry. Sunil is from medical biotechnology. And Sonia and Shiraj are again from psychiatry background. So all of us came together and did a set of studies where we were able to show that adding transcranial direct current stimulation helps to reduce treatment-resistant auditory hallucination. And over the past decade, this service has expanded, and we are running what is called as a Weiser neuromodulation program, that is weak intensity stimulation for enhancement and reintegration. And very recently, in collaboration with the engineering scientist, we have designed a cost-effective TDCS machine, which is very much useful for a setup like India, where we are in the process of experimenting home-based application. So this is the slide which shows how we train people for this home-based application. In parallel, we have also expanded these neuromodulation technique, and we are running a clinical research center, which is a multi-centered research program, which has a strong anchor on clinical research training. So essentially, if I have to reflect my learnings, perhaps the initial enthusiasm was triggered by the side effects, especially obesity, which made me to get into insulin and insulin signaling and insulin-like growth factor pathway, which made it plasticity and brain-related abnormalities. But throughout, the theme of my research was always guided by anchor to the patients and patient symptoms. And that kind of motivated us to get into this TDCS and related electrical stimulation technique. Now we have translated from lab to bedside, and now we want to move beyond and reach the community with this treatment. So if I have to kind of take this, I may want to simply say, perhaps the question was, is there a purpose for antipsychotic side effects? The side effects could be seen as a challenge, but many a time, if we get more into that, it will help us to understand some other aspects of the biology. So in that context, I would also like to, gladly list some of the publications which are from the colleagues here in the Department of Psychiatry. We have Dr. Ravi Philip, President Immunology works in psychiatry in collaboration with department here as well as CMC Vellore. Another interesting publication of Dr. Ravi Philip is this COVID-19 and mental health. So when all of us were grappling with the challenges of COVID, of course, he also handled those challenges, but he also gave time, thought through, and wrote this paper, which was published in the late 2020. I think immediately after the first lockdown. This was published in Asian Journal of Psychiatry, and this is a journal which I co-edited. And this is one of the highest cited paper. I was asking Dr. Ravi the other day, what made you to do that? He was just thinking through putting these things. So he had the vision to do that, and that has become one of the most cited paper in COVID and mental health domain. And also very recently, this is in 2023, this is a paper published by Professor Vikas Menon from here, who has just done a systematic review, which is not so research intensive, but this paper has been published in JAMA Psychiatry, which is the topmost list of journals in the field of psychiatry. So it emphasizes that if we want to do research, we can do at multiple levels, and we have the potential and opportunity to make bigger impact with less resource, as well as with, say, big science research, like what has been published in the area of autoimmunity. So to conclude, I would like to state that research is seeing what everybody else has seen and thinking what nobody else has thought. This one way of putting it across. In my view, the clinician scientist is more like a spectrum. There could be one end where one may want to do predominantly clinically anchored research, which could be just case studies and clinical database or N-of-1 trials. At the other end is the lab-based discovery science is something which one may be interested. A person can choose any position in this spectrum as per the interest and feasibility. While I agree that not all of us may want to become clinician researcher, nonetheless, most of us can add a bit of research to our clinics. And what is required is passion, curiosity, and perseverance. And of course, a great institute will enable, anybody that gets trained. For instance, a place like JIPMER has everything to a great clinician scientist. And I wish the students all the best. And with that, I would stop and acknowledge several of my mentors and collaborators. This is from the Individualized Schizophrenia Treatment and Reintegration Program. We also have a translational psychiatry lab. Students are coming from all background. And this is the Weiser neuromodulation team, again, students from different background. And then collaborators across the centers as well as faculty that work with me in the institute. I have to admit that it is primarily the students that do most of the hard work or all of the hard work. And I do the easy part of it in terms of talking about it. Thank you.
Thank you, sir, for your thoughtful insights.
We now request our director, Dr. Bhavna Badhe, to honor our chief guest with a token of our appreciation. Next, we request the dignitaries to release the Research Day souvenir, which symbolizes important ongoing research activities at JIPMER, traditionally presented every year during the Research Day celebrations.
Now, we have the presentation of the faculty awards. We would like to invite Dr. Sonali Sarkar to announce the much awaited results.
A very good morning to all. It is indeed an honor and privilege to announce awards for the best researchers and researches done in the last 1 year at JIPMER. So I would request our dignitaries to come forward to do the honors. The most awaited announcement, the award, is the JIPMER Faculty Research Award in Basic Sciences. For this year is being awarded to Dr. Rajesh Nachiyappa Ganesh, Professor in the Department of Pathology, in recognition of his contributions to the research in his discipline, the huge volume of work that he has produced. Congratulations, Dr. Next most awaited announcement is for the JIPMER Research Award for Clinical Sciences. For this year is being awarded to Dr. Adi Shivam, Professor of Neonatology, JIPMER. Hearty congratulations, Dr. Adi Shivam. We just heard the importance of collaboration from our honorable chief guest's keynote address. So to encourage more collaborative research within the institution, this year we instituted the Best Collaborative Research Award. And for this year, the award goes to Dr. Murugesan A. from the Department of Neonatology and Dr. Apurva Shankar Sastry from the Department of Microbiology for their work titled Clinical Profile and Risk Factors Associated with Elizabeth Kingia Anopheles Infection in Neonates Born in a Tertiary Care Hospital, a Case Control Study and Outbreak Investigation. Congratulations to the team, Microbiology and Neonatology. The next we have the awards for the postgraduate dissertations. The J.K. Datta Endowment Prize, which is instituted for the best thesis on infectious diseases for year 2025, goes to Andrew Mary Xavier from the Department of Pharmacology for his work titled Characterization of PNCA Mutation and Treatment Outcome Among Tuberculosis Patients. So congratulations to the candidate and the guide and the department. Andrew. The next award, Dr. Shyam Sharma Award for the Best MD-MS Dissertation, is for this year is being awarded to Dr. Kaustuba BG from the Department of Emergency Medicine for her dissertation titled Extended Cardiopulmonary Resuscitation Using Pediatric Self-Inflation Bag in Adult Cardiac Arrest Patients, Reaching Termination of Resuscitation Criteria, a prospective interventional study. Congratulations, Dr. Kaustuba. And congratulations to the Department of Emergency Medicine and Trauma. The next, the last of the awards is E. Mehalingam Award for the best publication by a PhD scholar. For this year, it is being awarded to Dr. Anuja Anandaraj from the Department of Pharmacology again for her work on molecular docking of curcumin and curcuminoids as a human zinc plus dependent histone deacetylase enzyme inhibitors. Congratulations, Anuja. Hearty congratulations to all the winners and the departments. Thank you, dignitaries. You can please take your seat. Thank you all.
With that, we have come to the closing stages of the inaugural function. I request Dr. D.M. Research to propose the vote of thanks.
Good morning, everyone. I feel honored and privileged to get this opportunity to propose vote of thanks on this special occasion. It is time to express and say heartfelt thank you to each and every one of you in this JIPMER team who contributed to the success of 10th JIPMER Research Day celebrations 2025. First, I have to profusely thank Professor Dr. G. Venkatasubramanian, Professor of Psychiatry at NIMHANS, Bengaluru, for giving us precious time for gracing this occasion and enlightening us with the words of wisdom regarding research through your keynote address. Thank you for clearing our concepts and enhancing our understanding. We have a great load of patients here. There is a great mass of people present who can be utilized for such purposes. Thank you thank you for you for this honor which you have been able to do for us. Next, I would like to thank our torch bearer of this event, our director. Many thanks to Professor Bhavana Bade, our officiating director JIPMER, for leading us today. Many thanks to the Professor Bir Singh Negi, our beloved director JIPMER, who could not attend this function as he was another assignment, for leading us, guiding us, making the changes this year to facilitate more participation and inspiring us in planning and executing the celebrations from the start to finish. We really owe you a lot. We also thank Professor Gururajan, Medical Superintendent, and Professor Dr. Bikram Pate, Dean Academy, for their guidance and support. Many thanks to the JIPMER administration. We have among us the Venkatasubramanian, the Deputy Director Administration, who have given unstinted support for the organization of this event. Our fantastic and vibrant team of Associate Deans Research, Dr. Suvita, Dr. Nishad, Dr. Prashant, Dr. Vikas Menon, who tried day and night to make this function a great success, and Tuesday with the help and support at every stage of this event, from conception to the completion. They really worked very hard and performed their duties diligently. Thank you all. At this junction, I would also like to thank our former director, Professor Rakesh Agarwal, for considering this research day in November 2024. The deans, medical superintendents, and my sincere thanks to the other dignitaries who have joined us on this occasion. We will ever remain grateful to you, and we will thank you for being with us today also. Thank you to the Scientific Committee Chairman, Professor Sonali Sarkar, and her team for awesome and fantastic effort during this edition of Research Day 2025. It was a research carnival of oral and poster presentation from undergraduate to postgraduate to the PhD scholars and non-staff, which was organized from 7th February 2025 to 22nd February. Each session was well organized by the scientific committee team and was well attended this year. An event of this dimension cannot happen overnight, and the wheels start rolling months in advance, and we have been fortunate enough to be backed by a team of very motivated and dedicated committee members and its members. Team worked tirelessly to make this function successful. Our organizing team, IT team, support team, volunteers, our admin teams, but true pillars and contributors selflessly in making this function come alive. The meticulous cooperation of the JIPMER team deserves special appreciation, and their support helped us to put this function in such a short span of time. Thank you all for everything. A special thank to Professor Chituria and the IT team for being huge support for conducting this edition of Research Day and transmitting it to the JIPMER Karekal, our other branch of JIPMER. Many thanks to all the Research Day committees members, venue and stage management, especially Dr. Lata and team, technical team led by Dr. Chituria and his members. From finance, Dr. Havasi, transport, Dr. Firiji, and prize and certificate, Dr. Ella Morgan, food and catering, Dr. Chatra, who always make the things lively, the good food, enticing the everybody to work hard. Banners from Dr. Gunaselan, and each and everybody who has contributed to the success of this. I may have missed out some names, but that doesn't mean that they have not contributed to its success. Many thanks to the workshop coordinators, the Professor Palnivel for an introduction to R for researchers, Dr. Mahalakshmi for qualitative research, Dr. Prashant Ganesan and Dr. Chakradhar for droplet digital PCR for health technology assessment. And finally, thank you to all the JIPMER faculty, residents, PhD scholars who have participated in this their research presentations and have been present in this audience as a part of the Research Day celebrations. Many thanks to the staff of the Dean Research Office and supporting research project staff for their support in making this event eventful. Last but not least, I would like to thank all the volunteers who helped in so many ways at every event, on every day of this event. We wish to thank you for all your wholesome support. Thanks to the photographer for taking beautiful pictures of our memories. In all learning experience for all of us, we will cherish the memories of this 10th Research Day celebration for a long time to come. Such events should serve to boost research in our institute. Let us all work towards that goal. I sincerely apologize if I missed out somebody. Thank you, one and all. Thank you.
Thank you, sir. I request everyone to rise for the national anthem.
जनगण अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड़ कलवंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण जय हे जय हे जय हे जय 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 हे
Would now like to invite all gathered to partake of the refreshments that have been arranged.
Refreshments for all the students have been arranged in the dining hall onto your left side in the connecting corridor. And for all the faculty dignitaries, please come to the ground floor MPH hall. And please make sure you be back by 10:30. Thank you.
I request us to load their PPTs for subsequent presentations to do so at this point of time. Thank you very much.
Welcome back, everyone. Now we move on to a much awaited session, one that promises to intrigue you all.
We rarely think about who puts the money in our bank accounts when we secure a research project. We're too busy celebrating after months of brainstorming. But maybe we should think a little more about our mysterious benefactors, the government or the pharma giants.
Should India rely on government funding, which amounted to 30 billion rupees in 2025 for health research? Or should the responsibility fall on the pharmaceutical industry, who contributes nearly 80% of research funding, despite its focus on profit-driven priorities?
To shed some light on this, it is our honor to invite our moderators for the upcoming debate, Dr. Srijit Parameswaran and Dr. Subhashini Kalyaperum.
I request all of you to kindly maintain silence. Let's listen to the debate. Thank you. Good morning. Good morning. I welcome all the esteemed faculty, students and friends who have gathered here for this very wonderful session. So it's indeed a very interesting session. In many conferences, debates are the most attended. And so we are here, and let's have a wonderful debate on two important topics, and let's have some fun with research. So I will just hand over the mic to Dr. Srijit. So he will introduce the first topic and the debaters for this topic. Over to you.
Thank you, Dr. Subhashini. Wasting time, we have very limited time. The first topic, as already mentioned, is the motion is that research should be primarily funded by government. We arguing on the motion. First is Dr. Usha Devi from Neonatology, who will be supporting the motion, and Dr. Shivdeep Ganguly from Oncology will be speaking against the motion. Before we start the session, a few ground rules. Each person will get 8 minutes. We'll stick to the time. Each person will 8 minutes to speak, and after both views, we will opportunity for rebuttal for 2 minutes for each of them. Then we will have concluding remarks from the moderator. Check. Hello. Yes, sorry about the interruption. I was mentioning briefly the rules. Each person will get 8 minutes to speak. This will be followed by an opportunity for rebuttal for 2 minutes. And then there will be concluding remarks. So we'll start with Dr. Usha.
Thank you, sir. A very good morning to all the esteemed audience, Dean sir and dignitaries and judges. So I'll be speaking in favor of medical research should be primarily funded by government, not the industry. So on the key points I'll be covering is like public health first, advancement of the basic and high risk research, equity in research, reduction of sponsorship effect, scientific integrity and transparency. So let me start elaborating point by point. So coming to the public health focus, as we all know, government research predominantly addresses the unmet healthy needs. It takes the health outcome of the public as a priority rather than concentrating on the financial gain. However, industry funding has a profit motive in all its research. And even 70% of them who were allotted industry research felt that there were too much emphasis on the applied research and commercialization of the product, and they did not take care of the humanitarian rights, and they were more towards the financial rewards, which is not ethical or acceptable. So this was from the scoping review where perception of the people who got industry support and those who got government funding were obtained. So now that we are too much into the social media, let me project some of the posts in the platform where since I'm from Department of Neonatology, we are exclusive breastfeeding. So it would be a sin if I don't project a screen which is on formula milk industry, which is favoring the formula milk and decreasing the breastfeeding rate. So one of the posts by the regulatory agencies like UNICEF India, it says that industry sponsorship, that is predominantly formula milk sponsorship, try to influence the health workers by means of gift, by means of grants for the research as well as travel. And this was a systematic review which was seeing about the conduct and reporting of the formula milk trials, where the most of the research was funded by the industry, this formula milk industry, and the results were the studies were having high risk of bias, and there was selective reporting of positive results. And another factors, so they included babies who were not on exclusive breastfeeding. So if we have such a scenario where the baby is not on exclusive breastfeeding, it is also our responsibility to enforce on a breastfeeding support to that mother. So today being a Women's Day, we should talk about breastfeeding support to the women as well in the postnatal period. However, this formula milk industry funded project did not take care about the breastfeeding support to the postnatal women and directly enrolled these babies into formula milk trials. And the outcomes also, there were selective reports, which led to high risk of bias. And this is, since Shubhadeep will be talking in favor of industry sponsorship. So here there is a post which is talking about a study where the pharmaceutical industry paid prescription of drugs by the physician and oncologist. So they were giving, rather than giving a branded drug, they were going for some low value cancer drugs, which might not be as efficacious as a branded drug. So, and coming to the second point, where the government funding supports the basic health research and groundbreaking discoveries. So, this government funded projects are more concentrating on the long-term research projects. They allow extension of the projects also if needed, and also their outcome has a long-term societal impact, and they concentrate on the public welfare. However, these industry-funded projects are driven by profitability, and they concentrate more on the quick fix solutions rather than concentrating on long-term societal impact. So investment in the rare diseases and preventive care is more by the government-funded medical research than the industry-funded medical research. So where they look for some trials, which will, where if they invest in those trials, they will get more profit following the outcomes. So next, government funding favors innovation and breakthroughs, whereas the industry sponsorship favors Me Too extension. Me Too extension means already there is a groundbreaking discovery. They make some modification to the discovery and they start marketing it. They do trials, they sponsor for that, start marketing it. So this is called as Me Too extensions. However, this government funding mostly supports innovations and breakthroughs, like the quick mRNA vaccine discovery without immediate commercial returns, and major medical advances like CRISPR editing, MRI technology, and cancer immunotherapy, where the majority of the funding came from NIH. And there is a proven track record. Like people say that majority of the drug trials are from industry. However, if we see the FDA approved drugs, NIH has made a substantial contribution to the FDA approved drug. And majority of some of the Nobel winning research are unfunded. However, majority of the Nobel winning researchers are funded by the government funding. So this picture, all of you will be familiar. This is a picture of Destin Banting. Banting went on to win the Nobel Prize along with McLeod in physiology and medicine. So they were working in the University of Toronto. So initially, the hypothesis that pancreatic extract might be useful in diabetic patients came to Banting. So he worked in collaboration with the University of Toronto. The initial fundamental research was supported by the government organization. Then he collaborated with other co-workers, and finally it led to commercialization of the product. One of the things of favoring industrial sponsorship, people say that from the time of hypothesis generation to commercialization, time is very less in an industry-sponsored trial. However, in this discovery, from the time of hypothesis generating the pancreatic extract might be useful to commercialization of insulin, it was a very short period, like 6 months to 8 months. So this is also possible with government funding. And in coming to equity, so if we see this left side, so disease burden, the x-axis show the disability adjusted life years or disease burden, and the y-axis shows the industry funding. So we can see that in high income countries, it is in alignment with the disease burden. Whereas in global disease, when we take a global disease burden, the funding of the industry is not in alignment with the disease burden. And it is not supporting diseases like HIV, AIDS, infectious conditions, which are more common in low and middle income countries. So they concentrate more on high market demand on drugs like antipsychotic drugs, which are having a chronic use, and overpricing and protected patents where they can overprice and they have more financial gains. Whereas if you see government funding, there is equal access and it ensures that medication, technology, and treatment benefit all people irrespective of socioeconomic status. So we can see that on the left side, the funding is in alignment with the disability adjusted life years. So accessibility to data and the product is more compared to industry funding. And sponsorship bias, it is a growing menace of recent times. So it can be either the influence in the design and conduct of the study. So we all know Tamiflu saga, where it was labeled as a costly mistake. There was a lot of lacunae in those trials, and the adverse effects like neuropsychiatric adverse effects were not seen at all in these trials. This is one of the trials where it was said that it was a double-blinded RCT, but clearly the capsule color was different between the placebo arm and the Tamiflu arm. And even in the Gardasil trial, though it is said as placebo, it also contained adjuvant. So there was some discrepancy between the protocol registered and which was the trial which was conducted. The publication bias, if you see, this is the line which is showing publication bias in the industry-sponsored trial, where there was a strong relationship between the positive efficacy and the publications, whereas the publication bias was less in government-funded projects and compared to industry-funded projects. So there was no commercial interest, and the transparent funding is there in government funding. Collective reporting is there in government funding. So fast tracking and expedited funding is also possible in a government-funded medical research. These are some of the programs which we can say which is in favor of expedited funding, which happened in our own country. So I want to conclude by using this Tirukural, which says that Payalum Malai Pol Pirandozhiginum Tam Viyalum Thanipadilar, which says that governors or a leader, rather than concentrating on financial gains, which is pouring like rain, we have to concentrate on selfless welfare to the public. Thank you.
We have overflowed the time. Sorry.
So very good morning to all. Thank you, Dr. Usha, my learned colleague and friend, for putting forward the fact that all the industry should not come into medical research at all. So our motion, which is in the floor, is that medical research should primarily be funded by government, not industry. With your permission, moderator, sir, I will be speaking against the motion. So if we go by my learned colleague does not believe in the vision of our Honorable Prime Minister. Our Honorable Prime Minister has recently instituted this Anushandhan National Research Foundation and conducted his first board meeting, and he has provided this vision of 50,000 crore corpus over next 5 years, of which the private source will contribute 72%. So this is the vision which we have for our country in the coming years. So whether our Honorable Prime Minister is wrong, that's what Madam believes, but we'll go ahead. So I will go by following points. My point number 1 is The funding research through government funding alone is not sustainable. Whether we like it or not, that is the reality, and it cannot build up the research ecosystem of the country. If we go to India's research and development expenditure as percentage of GDP, if we go to the first graph, which is on the left side, you see it is abysmally low. It went up, maximum never touched 1%. It went up somewhere to 2008, somewhere around 0.9%, and it is actually declining. And it is somewhere around 0.6 somewhere. And if we see among all the countries, here we are, China has surpassed us obviously for a lot of issues. The low middle income countries as a whole is increasing their funding. India, it is decreasing. So why this is so? If we go into actual research funding and try to categorize it, who actually funds? And if we see the list, and we all are familiar with these funding agencies, all are government. So if all are government, then where is the private research? This is the sector-wise funding of the research R&D in our country. And if we see the central government and all this, which includes actually the state sector and the public sector. So overall, this orange plus whole this part is contributed by government. So private is contributing only 36% in our country. Contrast to this, if we go to United States, if we see business is contributing 72%. So are they wrong? They are the number one producer of the research in the whole country, and the high impact publication wise China, even though sheer number of publication wise China has recently surpassed United States, but United States high impact publication wise continues to be number one for last 2 decades. So are they wrong? Yes, there is some concern that the bulk of this money is coming in the development. But development is also producing enough amount of new knowledge and innovation, which we should not ignore. So as a whole, if we go to our government, this is the budget breakdown of our government, which is the last financial year 2020, there is a breakdown. And if we see the major is in interest payment, subsidies, pension, giving the salaries of the government employees and everyone. So at the end of everything, we cannot expect government to come to our homes, feed us and everything. So we cannot also expect our government to take shoulder all the burden of research in our country. The government fiscal situation is unlikely to change anytime soon. But if we have to improve and progress as a country, we have to private investment and private also companies, and depending on government is not sustainable. So why the private involvement is? Sorry. So coming to my point number 2, the private R&D is more accountable and likely to achieve goals faster and more efficiently. So as Madam was pointing out that there has been 2 examples like insulin and also COVID vaccination. But is it really? Is insulin currently being produced by government organizations? They're currently not being produced by government organizations. They're produced by actually pharmaceuticals. All the newer generations of insulin, who have discovered? Government organization? No. All the newer generations which we are currently using are manufactured by pharmaceutical industry. It was year 2000 when Bill Clinton was standing along with Francis Collins. The Francis Collins, and this is the CEO of Cellular Genomics. It was Human Genome Project. At that time, Francis Collins, head of NIH, was leading the Human Genome Project, trying to produce the first draft of human genome. And Cellular Genomics wanted to do it parallelly at the same time. They were racing together, and Cellular was almost going to publish its genome faster than the Human Genome Project by the government setup. And the White House was facing an embarrassment. So they went ahead, even without completion of Human Genome Project, they brought them together and announced that we have actually completed the Human Genome Project when it was not. The race was already lost 2 decades back, but Madam wants to go back 2 decades. So everyone, I think, recognizes this person, which is Elon Musk. This is the cost of space flight, which was there when all the government organization was doing space flight. And this was the cost per astronaut, which was used to be taken by the government organization to send an astronaut to space. Elon Musk, when SpaceX was founded, For 10 decades, for 1 decade they put forward, went nearly bankrupt. But now today, the cost of spaceflight has come down. And from 2012, NASA actually lost their capability of sending humans to space by American spaceflight and was entirely dependent on Russia. Only with help of a private organization, United States astronaut can travel today to International Space Station after they have discovered another Falcon 9 rocket. The COVID-19 vaccine development is a private endeavor. We believe that government has funded, but government has not funded actually. It is most of our own pharmaceutical industries in India who have funded developed vaccine, which is Bharat Biotech, obviously with some input from ICMR, but predominant investment has come from the industry. The mRNA vaccine, we have forgotten, India has also generated by private investment as well. My point number 3, as Madam tried to find out that high-risk long-term funding is not feasible to private funding at all, but it is feasible actually. A big tech, for example, Microsoft, Google, IBM, they have been funding artificial intelligence. See, this is from 2000 to 2016. At that time, AI was not being heard, not being discovered, I'm wrong, not being in the public discourse. But they were funding at that time. And at that time, the companies, without any appreciable inputs that whether it will be profitable even, they were giving that much amount of money to AI. Google has been doing it from 2012 for last 1 decade. And that was already been with a vision of long-term funding, even without appreciable input that whether it will become profitable or not. In United States, average duration of a biopharma and biotech startup to be profitable is 7 to 10 years. And in a routine pharmaceutical industry, for any molecule to come into it, they need more than 1 decade of investment and around $1.7 billion. So do you think that private organizations don't have a long-term vision? Do you think that private organizations are only bothered about their quarterly profits? If so, then why are they funding for decades? And mind you, all those molecules actually, even after all the trials, do not actually come to market, and the research goes to a lot. And the blue sky researches, which are the curiosity-driven researches, are also funded by pharmaceutical industries. Amazon, for example, is giving a funding for childhood research, which I am sure you will be very happy to apply. But yeah, those are private, mind you. And obviously, Blue Sky Initiative by Michigan Engineering also being funded by Google. So private, not bad. Private does not mean
your
bad.
time is up.
And when led by visionaries, it can be an important source of long-term innovation. So as I mentioned, my 3 points, and so it is not sustainable. Private funded research likely to be more accountable, efficient, and high risk and long-term research innovation is feasible to private R&D also. So maybe the vision of our Honorable Prime Minister is right. Thank you.
Thank you, Dr. Subdeep, for your very convincing arguments. I would like to invite Dr. Usha for her rebuttal. You have 2 minutes.
So as Shobdeep pointed out, so private funding might be by non-government funding. Here we are talking about not the NGO funding. We are talking about exclusively government funding versus industry funding. And we are not taking NGO to bring clarity to this. We are not talking about funding from the other NGOs or private institutes. We are purely talking about government versus industry funding. And then regarding a point where Shubdeep told that development can happen in the more in the industry sponsored trials, we need to remember that I previously told regarding insulin discovery. So it all, like our chief guest told, it all depends on the motivated people with perseverance. If they are present in the institute, it is possible even to create such groundbreaking discoveries, even in a government institute through government funding. That is one of the points. So, and then we have to remember that insulin initially they patented because the idea should not be taken by other industry or private organization. and it should not be patented for high cost, and it should not be made unavailable to poor people. So it was patented with the University of Toronto for $1, which is very less. Suppose if it has been founded by an industry, it might have been patented for a higher amount and it might not be available to all those in needs. And so long-term funding, yes, it might be possible even with industry sponsorship, but we need to remember some influence on the outcomes, and there can be some profit motives in those as well. So that's what we are talking about, a biased research, which can happen more with the industry sponsorship than the government funding. And then even Shruti might be having some conflict of interest. He is part of big projects funded by government, where even they allow phase 1, phase 2. They are funded for phase 1, phase 2 trials for drug delivery. So
thank you, Dr. Usha. I think your
Thank
time
you.
is up. Thank you.
Thank you.
Dr. Shubdeep, would you like to rebut her arguments?
So overall, the main point which Dr. Usha is trying to focus on, that private means profit, profit means bad. We should not think profit is bad. All of us would like to have more money. So as long as they are willing not to, play with the ethics, and the playing with ethics does not differentiate between public and private. A conflict of interest can exist even in a government setup. A person who is not ethical may exist both in public setup and in private setup. So ethics is never the point where, for example, in a private setup, if a visionary is there, a long-term funding and a vision is there, and its ethical person is there, be more efficient, they can actually produce more innovation than an area where there is more inefficiency and more bureaucracy is there. That is my overall rebuttal to you. Thank you.
Thank you, Dr. Usha Devi and Dr. Shivadi, for that excellent points of argument for this debate. So just to summarize the points, for government funding, Dr. Usha Devi brought out that it promotes research based on public health issues, and it's more transparent. Actually, the data, when it is government funding, is easily available in the public domain, and it's cost effective, and there is accountability to the public. But then when you see the downside, which Dr. Sivadeep also brought out, that it is very slow to materialize. The funding rules are also rigid, and actually they focus on certain diseases and not innovations. And it's the funding also is insufficient. So and it's not sustainable, as you said. So when it comes to industry funding, it drives innovation, and then it's more efficient, and they have clear goals and guidelines. And the industry funded research, they take bigger risks. And the resources also, as Dr. Pradeep said, the funding is much better with industry. And he gave the US example versus India, where the US funding is more industry funded research. And but then the main point for contention, it is all profit driven. So that is, there's no denying that they focus on more profitable areas than, and then they neglect the broader public health needs, which are essential. And it's more biased. There are some industry driven research which manipulate the results, and sometimes there's lack of transparency, and they do not disclose the methods and the results. And as Dr. Usha Devi told in a rebuttal, sometimes they are all patent protected, and there's limited accessibility. So not many can afford those kind of drugs or other things, devices which come into the market. So, but then again, having said all this, significant medical breakthroughs have happened, have more often happened with industry based funding. So with all these points being considered, so I think I firmly believe that I think we can have a balance of both government and private industry funding. We should look at options of this private public partnerships or some other private public models which can balance both the government and the private interests. And so I think with these, or if it is purely industry funded, maybe we can ensure, the government can ensure that the regulations are much stringent and they take into and they consider research based on the public health needs. So with these final thoughts, I think we can move on to the next debate. So the topic for the well done and congratulations to the debater. So the topic for the next debate under the resident category is artificial intelligence tool to support dissertation work should be allowed. And we all, and this is actually quite an interesting topic now that residents have submitted their thesis dissertations. So artificial intelligence has indeed become integral. Right? So smartphones, smartwatches, you can see AI is there everywhere. So why not in dissertation? So that's why, I mean, so let's hear from the young, dynamic residents who will be debating on this topic. For the motion is Dr. Anindo Mitra, junior resident in the Department of Psychiatry. And against the motion, that is against artificial intelligence, is Dr. Gitangshi Gopal Gabru, junior resident in surgery. So we can start off. So,
a very good morning to everyone present here.
just a minute. I just want to reiterate the ground rules. You have 8 minutes for presentation and 2 minutes for rebuttal.
Very good morning to everyone present here, respected moderators, faculty members, and my fellow participants, and the audience. It's an honor to stand before you today on 10th Annual Research Day of JIPMER to discuss a topic that is shaping the future of academia and research. I'm Dr. Anindo Mitra, a junior resident in psychiatry, and I strongly support the motion that AI tools should be allowed to support dissertation work. We live in a time where technology is transforming research and education at an unprecedented pace. The question is not whether AI should be allowed, but how we can use it responsibly to enhance the academic integrity and efficiency. In the next few minutes, I will present why AI is not but a valuable tool, one that improves efficiency, promotes fairness, and prepares the researchers for the future. Imagine a world where a postgraduate student has an intelligent research assistant available 24/7. An assistant that organizes literature, refines writing, and ensures clarity, allowing the student to focus on what truly matters, original thought and critical analysis. That world is not a distant future. It is here, and it is powered by AI tools like ChatGPT, Grammarly, Zotero AI, and ResearchRabbit. The question before us is, should we embrace this progress or reject it out of fear? A dissertation is not just about writing. It is a marathon of research, revise, AI can automate repetitive tasks like citation management, formatting references, summarizing lectures, summarizing literature, and even detecting inconsistencies in writing. AI research assistants like ResearchRabbit can drastically improve the efficiency by quickly finding relevant literature and even visualizing connections between the papers. It can visualize not only the papers that have been cited we're looking at, and also for the future papers that are cited the particular paper. It can also at one glance find out all the research that has been done on that particular topic and by the particular researchers. Not only something that would take us about 6 to 7 hours can be done within like 30 seconds. Likewise, citation managers such as Zotero, now exploring AI integrations, can help organize sources and format the references automatically, saving hours of manual work. Overall, these tools act as research force multipliers, handling routine tasks with speed and accuracy that humans alone cannot match. Grammarly offers real-time grammar corrections and style suggestions, acting as an ever-ready proofreader and editor. This is especially valuable for students who may not be writing in English as a native language. AI can help them maintain their voice while achieving clarity and coherence. Would we tell researchers to avoid search engines and go back to sifting through physical archives for months? No. AI is simply the next step in the evolution of research assistance. A 2024 systematic review found about 24 studies found that AI was used for enhancing idea generation, content structuring, literature synthesis, data management, editing, and ethical compliance. Not all students enter postgraduate studies with the same advantage. Some struggle with language barrier, while others lack proper writing mentorship. AI democratizes access to high quality writing support. It levels the playing field, ensuring everyone, not just students, institutions, can produce well-structured professional dissertations. Writer's block and organizational challenges are common in dissertation work. AI tools can serve as creative partners to overcome these hurdles. AI can help structure complex research arguments by providing scaffolding onto which the research can build on. Is it fair to disadvantage students who don't have access to expensive tutoring or research assistance? AI is not unfair shortcut. It is a tool that allows students to express their ideas more effectively. These two 2023 studies done in Australia and Spain respectively reported about the benefits in native English speakers. Residency is a full-time clinical role. Unlike PhD scholars, residents balance patient care, emergencies, and research. AI can help manage this workload by cutting down tedious non-academic work. Formatting tables, restructuring paragraphs, revising grammar, these are the tasks that AI can handle in minutes, saving hours of efforts. AI allows the residents to use their time where it matters the most, on clinical practice and original ideas for research. Would we tell scientists to stop using statistical software and go back to hand calculations just because it automates the process? No. AI does not generate novel scientific discoveries. It helps organize and refine the ideas the students already have. A well-crafted dissertation still requires critical thinking, hypothesis formulation, and methodological rigor. AI is no different than the advent of Microsoft Word, spell check, or statistical software like SPSS. These tools did not make research weak. They made it more efficient. AI should be a guide, not a replacement for understanding. The key is responsible use, ensuring AI remains as an aid and not a replacement for intellectual effort. We must embrace AI wisely and not ban it blindly. This recent 2025 cross-journal analysis reported that the majority of the AI that was used was to improve the readability and to check the grammar. AI is already shaping the future of scientific research and publications. Major academic institutions are integrating AI-driven tools for literature synthesis and data analysis. Banning AI in dissertation writing isolates the students from real-world research trends. Should we train researchers with outdated methods only for them to enter a workforce that embraces AI? Instead of prohibitions, institutions must focus on AI literacy, teaching students to use AI responsibly, ethically, and effectively. What comes to our mind when we talk about allowing use of AI for dissertation, instead of what it actually means? Can you help me understand this SPSS output? Can you help me create a table summarizing the results? I have 36 articles to read through. Can you summarize all of them in a tabular format? How can I reframe this to make it more concise? Concluding, AI does not replace the intellectual rigor for dissertation writing, it enhances it. It saves time, reduces unnecessary workload, allowing the student to focus on research and critical thinking. Rather than rejecting technological progress, we must harness it responsibly. Universities should set guidelines for ethical AI use, ensuring it remains as a tool for empowerment and not dependency. If the goal of education is to prepare students for the future, then embracing AI is not an option, it is a necessity. I urge you to support this motion. Let's embrace AI as a partner in academic excellence. Thank you.
Yeah, so wonderful argument by Dr. Anindra Mehta. So we'll go for against the motion by Dr. Gitankshi Gopal. She doesn't have a PPT, so she's going to speak. So, but still, you have only 8 minutes.
Good morning, everyone. Fellow very beautifully presented the points on why we should favor AI. But here I am against the motion for that it should not be used for dissertation writing. As we all know that dissertation writing plays a pivotal role in essential component of research, which is characterized by structured expression of ideas, data-driven arguments, and logical reasoning. The whole point of including a dissertation writing in the postgraduate program or in the is for development of research skills where people actually come up research, look for matter where there is lack of research, where new development is required. They identify those research gaps, then work on it, bring up a plan for it, make a hypothesis for it. Make a hypothesis for it, and then they generate the data, collect the data, analyze it. No doubt AI can be used as a tool, but are we responsibly using it? Do we really know whether we can actually identify, and how can we identify on what parameters that a student will use it very responsibly, bringing up only some certain points of only statistical analysis or any editing per se? Or how is there any parameter available to identify that? As my fellow opponent very clearly told it, do we really need to rely that much on artificial intelligence and not believe on our real brains? Though AI is revolutionizing academic writing, it poses challenges in academic integrity as well. As for me, academic work should maintain its authenticity and depict the intellectual labor of the writer. Do we really need to depend on the bot-generated assignments for understanding our assignments and to simplify these articles? I firmly believe that AI should not be used for dissertation work and writing. Following are my points, as it undermines the academic integrity. There are increased risk of plagiarism as well as lack of originality, as academic integrity requires students to submit their original work in the form of their own speech, analysis, and thinking. Use of AI to generate any significant portion of the paper also can lead to unintended plagiarism, as AI mainly works on data and patterns, and it identifies common sources and ideas without a proper attribution, leading to plagiarism, and this might pose further issues later, undermining the original thought required for the academic work. There might even be misinterpretation of the effort and the knowledge, as student might over-depend on it, on the AI, and bypass the intellectual labor to produce the academic work, and also undermine the research rigor, as it is easier to actually open a ChatGPT and ask for what new latest thing I can think about, rather than sitting and brainstorming my brain over like what new idea should I work on and what research should I do on. It's easier to at a click on the phone. Currently on WhatsApp also we open our WhatsApp ChatGPT, we can open it and identify what new research can I think about it. Should we really rely that much on artificial intelligence? Also, there may be fabrication of sources or citations. As accurate citation is a cornerstone of intellectual honesty, AI-generated content sometimes creates a very convincing narrative, but without any valid sources. A phenomenon known as hallucinations. It was described in one of the Elsevier studies, where they told that AI, when they had done multiple sources and channeling how papers were generated, they very beautifully created a very nice paper over it, citing some articles which were never ever done. Thus, this would first mislead the reader, and it would also violate the citation standards. And in addition to that, if you're talking about that it is helpful in the students, there are certain people who are not very well versed with the AI world as well. Some people might be struggling reading and would prefer the conventional way also. Don't you think that would also pose a threat and cause a academic demerit? For someone, there are two aspects to it. One, if he gets an upper hand by using, he beautifully created a dissertation, gets more marks, then that it is unfair and unjust to the person who has been actually conventionally trying to produce this paper. Second, it also could also lead to, if identified and not used adequately with proper following the rules, it may also lead to expulsion of the student, or it might pose threats to his degree as well. In addition to that, it also hinders the learning and the skill development, as critical thinking and analytical skills are important, because usually it is easier to, it bypasses the mental effort to analyze, evaluate, and synthesize the info, and it hinders in creativity, as it is easier to rely on the AI prompts to generate a new idea rather than sitting and brainstorming on things and increasing our boundaries of imagination. In addition to that, it also weakens the writing and the communication skills, which during the course of dissertation writing and the editing and the refining, and this refining happens through various feedbacks that we receive from our colleagues or from our professors. Also, it stifles the collaboration as well as the peer learning as well. As if we rely on the AI to do every work for us, including editing and all, why would I actually go to my professor and ask for some valuable info? I firmly believe that their years of experience would give me a better outcome than relying on an AI. Accuracy issues because we do not have verified sources or citations. Also, it might not adapt to the specific writing styles that are planned for a particular dissertation depending on the institutional policies. And there is a lack of context and nuance because the references sometimes that are produced by the AI are superficial and are correct on the surface, but they do not actually delve deep into the implications of the study. It even poses threats to the academic standards, devaluing the need. Why do we actually write a dissertation for the degrees for postgrad programs, as it would no longer reflect an individual effort. So I would conclude saying that let's not just rely on the artificial intelligence and would think on the real intelligence, our brain as well. And an author needs to be someone who can actually provide consent and affirm what they have followed the ethical protocols of the research and they did steps as they said they would. And to do that, I think so you need to be a human rather than an AI or a bot. Thank you.
Thanks, Dr. Gitangshi. You're finished well ahead of time. Yes, Dr. Anandu Mitra, you have 2 minutes for rebuttal. Just one question. Did you use AI for your PPT preparation? No. Okay.
I anticipated some of these things to come, so I prepared slides for this. Like coming to the first point, one of the things that she mentioned was AI reduces originality. However, when we speak about this, it fundamentally misunderstands what AI is supposed to be used for. AI tools are fundamentally reactive. They provide assistance based on the prompts and the existing data. They cannot originate true academic insight or deeper critical analysis on their own. So the researchers must direct AI, choose what to ask, and crucially, interpret and build upon the AI output. The intellectual heavy lifting, the idea selection, the hypothesis testing, interpreting the results remains the student's job. Coming to the fact that AI increases plagiarism risk, more importantly, firstly, using AI ethically is technically not plagiarism. Universities have already provided guidelines. AI generated text without any attribution is plagiarism, but using it with proper citation is not. Rather than promoting cut and paste writing, AI tools can act as plagiarism safety net. For example, Grammarly, it flags potential citation issues on overly closely paraphrasing. She mentioned something about hallucinations. It is very true that AI models can sometimes information, which is called as AI hallucinations or reflect bias. This is a valid caution, but not a fatal flaw. The argument for this is oversight and not ban. Academic research already demands skepticism and verification of all its sources. Whether a student finds a claim in a journal article or gets one from AI, they are expected to verify it. AI is no exception. Citations must always be cross-checked. And as for the fact that AI weakens writing skills, the key here is balanced integration. History shows that whenever new tools emerge, education adapts rather than it deteriorates. For example, calculator. Initially, it was feared, but now it is essential for advanced work. AI, like calculator, frees the student to focus on complex problem solving. And two other points you mentioned that not everyone knows how to use AI properly. I like to point out that ChatGPT can teach you how to use ChatGPT properly if you know what to ask it. And if another thing
Your
that
2 minutes is over.
one last point. You mentioned that what is the point of going to professors if ChatGPT can correct your grammar? I really don't think the role of professor is to make corrections. They have a much deeper role in our dissertations. Thank you.
Over to you, Dr. Vitanshi, for your 2 minutes rebuttal. Did you purposely not prepare a PPT? Yes, because
It was not a way we could prepare. Putting forward what he said is like we can use a chat GPT to actually ask for using how to use a chat GPT. Do we really know that everyone in India actually has a phone right now where we could use a chat GPT? In addition to that, professors per se is mainly going for professors like to know mainly from the experiential learning for over the years that professors have given us. They can give a valid input to each and everything that we are doing, whether the writing of dissertation. See, dissertations per se are for our academic development. It put forward us ahead in academic development for a new career or a new place that we are going to. So for writing for that and learning the entire skills and all that we will hone after going through this rigorous process, I think so we should actually rely on it. If we are using responsibly is a separate thing, but who is defining that responsibility and on what are the check parameters currently that the AI can be used? And currently, JIPMER is having any AI regulations available that how many people over here would have actually submitted their dissertations and used an AI and no one would have actually known? Is there any currently policy available that could actually tell that this person has actually judiciously used an AI tool and not actually overused it? In addition to that, he might get an upper hand also in the academic, getting an extra marks, getting an extra arena. I personally feel that we should keep certain things the original way, leave it as they are. Some amount of help from internet we can anyway get, which was internet sources were there. Now we are not delving into through the books, looking for things. We have internet available. But really relying on artificial intelligence, I don't think so. Too much is helpful.
Thank you, Vitanshi. I think we have listened to both sides of the argument. Briefly to summarize, one of there were 7 main points put forward against the motion. One was that it will impair the abilities of the learner, that they will not acquire the skills. Second is that the authenticity or originality of the work may suffer if you use AI tools. And there is questions about integrity of the material, integrity of the work, as well as the chances of inadvertent plagiarism from using what AI has given. And the very, very valid point about the output may not be reliable, which is actually true. And again, some people may not be able to access AI, which may not be a real issue in the current era with a lot of availability of mobile and other things. And emphasizing on responsible and ethical use, he highlighted that we are probably not having a regulation as of now. So that is a typical way where regulation usually lags behind scientific development. So that catching up has not happened. So that is one argument against using AI tools she was telling. But the supporting, there were I think I would say there were 5 arguments. One is that main argument is that it is not about replacing our ability. It is augmenting or supplementing our ability. Our cognitive abilities and our analytical abilities are not supposed to be replaced. A responsible use is to supplement our or the learner's abilities. And other was about improving. Some people may not have access to AI, but AI may actually increase access to, say, a typical example is language model, which he mentioned. Impaired language ability may be compensated by using supplemental AI tools, which is a very valid argument. But again, this has to be responsible use, it has to be ethical use. So putting together, I think it is evident. There are two additional things which I think is relevant, that we are already using a lot of AI. We are using a lot of technology for many years now. And nobody will argue that we should not use aeroplanes, we should still travel by train, or is a more advanced technology. I would think it is appropriate to say that cognitive abilities, our analytic abilities should not be compromised. That has to be there. You have to learn under a professor, under somebody's guidance, but AI may help you to take you to the next level. You will augment it may improve your efficiency, it may improve your work quality at the end, but there has to be limits to there has to be regulations. It has to follow regulations. It has to be used ethically. If we use it for the right purpose, I think we can safely say that over and above human abilities, you will be able to achieve more with the help of AI. Thank you.
That definitely gives us some food for thought. We'd like to thank our moderators and our speakers for such an engaging session. As we move on to the next segment of today, I'd like to call upon our Associate Dean's Research, Dr. Prashant Ganesan, Dr. Subita, and Dr. Nishat Plakar, to tell us a little more about research in JIPMER.
Morning, everyone. Thanks for being here. Over the next 20 minutes or so, we just want to take you through the activities of the research division of JIPMER. So we'll just quickly move on to tell you what happens behind all that, behind the scenes in the research division of JIPMER. Involved in many of these activities, so you would be aware of most of these things. Just wanted to give you an overview. This is mainly directed against the young faculty who have joined over the last couple of years, for them to understand the processes of JIPMER and process of research in JIPMER. Few of us are here. So Dr. Vikas is not here. So first I'll talk about the organization and activities of the research division. I'll just give you an overview. More specific aspects will be dealt with by Nishad and Subita. So we have the following goals of improving the standard of research in JIPMER and trying to increase the extramural funded research in JIPMER, and also trying to make the overall atmosphere more friendly for research and ultimately enhance the quality of our research publications. So with this, within the research division, we do management of various committees which are related to research. We organize various workshops throughout the year, some of it which was done in the last 1 month as part of the research day, and also throughout the year at various time points. We have annual research activities like the research day reconnaissance by the undergraduates, which happens next month, and the research course, the administration of PhD courses, the PG dissertation, and various research-related softwares which are currently in use, like Grammarly, SPAX, and Authenticate. The administration of intramural and extramural funded projects, which takes the bulk of the work of the research division actually. And in addition to the various funding agencies, we also help facilitate international funding in the bio-RRP portal facilitation, DSI, and also facilitate the conduct of sponsored clinical trials. There's some additional work like administration of PNMC credit points, and also periodically providing research-related data and statistics to the government bodies. So this is the organization of the research division, again, for mostly for young faculty. We have the research office within which we have the research division office, the research finance, which is in the admin block on the 3rd floor, and general grant facilitation. The various committees, scientific committees, ethics committees, and some regulatory committees. The research infrastructure, which includes the central research facility, the animal house, bioinformatics facility, and the management of the various softwares. We are going to have some discussions today on these specific facilities which are available, what they can provide for you. Dr. Rajesh and Dr. Meda are going to talk to you about it, so I won't be covering much. We have a central research facility which is housed in the JIPMER building, which started from 2020 and has now grown to provide more facilities. So how you can use these facilities will be discussed later. We also have a bioinformatics facility which started a couple of years back. Dr. Meda is managing that. This is in the e-learning center on the 2nd floor of the JAC building. We have a central animal house, which is now functioning as a fully separate building. Animal research facility. I think just 3-4 months back, we had a full session on this as part of one of the research circle talks. So it has a centrally air-conditioned with an HVAC system. There's space for different amenities like imaging, small animal breeding, facilities for drug development, toxicological testing, laboratories for teaching and training for different departments, and more advanced facilities are also planned in the next phase. These are the various committees which manage research in JIPMER. Some of these are necessary for the scientific reviews, like the UGRMCs, JSAC, PhDRMCs, sponsor trial committee. And we also have the regulatory committees which are necessary for granting the ethics approvals for various projects. There are some other committees which are necessary for managing the various workflows. Over the last 1 year, we have taken some steps to make research easier. So one of them is we have put out recruitment guidelines, especially for extramural projects. We have specific guidelines for DSMB constitution, which type of studies require DSMBs. We have SOPs for conduct of sponsor trials. We have specific processes for facilitating research-related agreements. Now we have simplified the whole process and it can happen in 1 to 2 weeks now. And we have also simplified processes for retrospective studies and case reports. We are also planning to simplify some more procedures here. We have unified submission processes for all committees, which is coming up. We also planning to come out with guidelines, documents for procurement related to research and simplifying the workflow in the research finance. There are certain new initiatives which we have done in the last year. We have an MOU with ICMR through which a young faculty PhD program has started, and we are happy to say that 5 junior faculty from JIPMER were chosen for this program in 2024. And this year, the director also called for new innovation, incubation, and entrepreneurship cell. So this will hope to promote development of innovative ideas and products to enable partnerships with the industry. So with this, I hand over to Subita for continuing.
Thank you, Dr. Prashant. So I'll be taking over and talking a bit about how the management of research projects are undertaken in the Division of Research. So currently, we look at the landscape of research opportunities. For all stages, we cater to starting from undergraduate research to postgraduate research, doctoral research, and research. So the nature of funding is non-funded and intramural funding as well as extramural funding. And so they range from single investigator initiated to multicentric, multidisciplinary collaboration studies. And there are more number of studies that are coming up in the past few years in this regard. And the types of extramural opportunities that are available again are of a varied kind. And now ICMR is more streamlined in this process and many other organizations as well. So there are calls for grants, task force related applications, expressions of interest, advanced center calls for, and other special calls for. So just to give a brief overview of how we conceptualize the life cycle of a research project. So it starts much before the approval comes or the funds come in. So the first concept proposals are written with years of experience. And then the initial budget conceptualization also needs to take place. Coded documents have to be submitted once the approvals are obtained for the projects from the extramural funding agencies. And then the other approval process starts within the institutions with respect to scientific approvals, ethics approvals, various administrative bodies as applicable, MOUs as needed between the partnering institutions, regulatory approvals, and other HMSC for international funds. So, and then starts the initiation of the project. So where the accounts are mapped in our SFAX, that is a financial software that we have right now. Manpower recruited, and then along with it happens a purchase of equipment and other consumables. So along with it, once the manpower are in place, so data collection happens, simultaneously bill submissions, and then comes the periodic reports. And annual reports are submitted as well as UCSOE to be generated and final project submissions to be done for the funding agency as well as other publications. So the research division submits in all these phases, and we are still trying to streamline the process so that there's less efforts required from the PI side in this regard. But of course, there are several versions or iterations, modifications that happen with the funding agency in terms of financial mechanism and others. So it takes some time for us to adapt to it and to be responsive to the changes needed. So these are the various places where we have tried to bring in some order and this in the last few years. So, and for the recruitment, again, we have a guideline in place. So in that guideline, we have clearly now unified type of applications. So where the investigator sends the request to the Dean Research Office, and once it's approved, it is advertised in the website, and then the process of recruitment happens, results are published, and then the man into the financial software for the process. So this is what we talk about in terms of SPACs, that is a fund-based accounting solution from Stira. So they've been a steady support for the past 5 to 8 years for the institution. They conduct periodic trainings for the research staff as well as for the PIs on a couple of occasions. So we intend to keep this more frequent so that the new faculty also get familiar with this process. And now our finance division is expanding to keep up with the increasing projects. And therefore, maybe even we'll be streamlining it more in terms of further work desks and help desks. So these are more extensive details about the modules available in this software. Finally, about some of the challenges that still as PIs we face on a day-to-day basis are delays in UCSOE, frequent changes in the funding mechanisms, GST related issues. So based on how or what type of projects we undertake. So we do have some newer mechanisms that are in the pipeline. So looking for additional manpower for processing files. So this one more thing is about bank reconciliation that has started in the last year. So many times we are not aware when the funds come in from our funding agency. So this process has really helped in that we are able to inform on a real-time basis that these are the funds available. The PIs can start the mapping process immediately. So the other one is about the research overheads. So we had a process earlier where 40% of the overheads money that the PI brings in goes back to the researcher for enhancing the research. So again, that has been more streamlined and mapped into the research software. So where the institutional overheads are further channeled into the efforts, for example, for the manpower, the additional manpower that we have right now for all of these processes we said. And the 40% goes back, and we have some guidelines in place how the PI can use it in a more structured format to enhancing research for procurement of a few small items or for publications and other types of needs. So purchase guidelines are still in place. Then these are frequent capacity building workshops that are conducted by the Division of Research. And we also hold the PhD capacity building program with 5 modules. So it's all by a collaborative effort with the various faculty involved in all the modules. So this I hand over to Dr. Nishad for the briefing. Thank you.
Good morning. I'll talk about our research funding and support and how it has changed in recent years. We have intramural research funds primarily for trainees, but more recently also more for faculty. So for MD/MS students, we provide 2 lakhs per candidate, up to 2 lakhs as funding, and same for DM and MCH. For PhD, that goes up to 7.5 lakhs. And for postgraduate students, about 50,000 rupees. And these amount to approximately 2 crores a year in the last few years. That's approximately how. In addition, there are the JIPMER Awards for undergraduate research. Last year, there were 26, and this is going to increase to 40, and there is an increased focus on innovation. So 10 out of the 40 projects are going to be exclusively focused on innovative ideas. We have a total of around 2 crores dispensed for students, as I mentioned. And the bulk of this goes to MD, MS, and MDS students because they are a much larger number than the other groups. We also have more recently instituted an early career intramural research grant. This is for faculty members who are at the assistant professor level. The maximum amount is 5 lakhs. There is some room for changes in that too. And this is funded for a period of 2 years. The priorities for the research project are the award is based on the scientific merit of the proposal, feasibility, applicability, and especially collaboration with other departments. The number of approvals is flexible. There is no fixed number. So if there are enough projects that approved, will be approved irrespective of the number. And another new thing that was introduced more recently was the competitive intramural research grant. This is for all faculty, permanent faculty who have at least 3 years until superannuation. 6 lakhs funding for a period of 2 years. Again, the same priorities and flexible number of approvals. So those are more recent, but the bulk of our research funding actually comes from extramural research funds. And we have been doing fairly well when it comes to extramural research funds. In the last few years, our research funding has increased significantly. So you can see here, these are the funds actually received. This is not by financial year. This is by financial year, sorry. So this is the actual amount received by JIPMER from extramural funding agencies in the last 3 financial years. So you can see in the last year, this has been the highest ever that we received, around 27 lakhs. Overall, we have just around 200 funded projects running right now, and with a total sanctioned amount of about 107 crores. So the research funding that we have attracted has been increasing over the last few years, and quite significantly so. The bulk of the funding comes from public agencies, and you can see ICMR is the biggest of those, and then DBT. But also, what you can also see is that there are a large number of funding agencies now. As of last year, attracting funds from about 60 odd different funders for research. Some of them only funding 1 or 2 projects maybe, but wide variety. So our funding base has also increased. Amounts have increased. The number of projects have also increased quite significantly over the last few years. We have also, I think Dr. Prashant and Dr. Subda already covered many things that we have done to make research easier in JIPMER. And the research web pages have also been updated, and it has been revamped quite significantly to make a lot of information available for, especially for junior faculty who are not familiar with the processes for research in JIPMER. So you can see on our main website under the research heading, if you go, you can navigate to all the resources that are available. I'll just show you some of the examples of information that is available on the website. Extramural grant related links. So if you want to look at the funders Those links are all provided on the website, so you can go check yourself periodically to see about calls for funding from these agencies. There is a whole web page on the various research guidelines and submission guidelines for research, because there are so many of them now. You will need some help, I think, to navigate those, and that's provided in the website. You can also find for specific research grants, if you need to look at details, those are also available on the website. And that, I think, is a broad overview of what has been happening in JIPMER over the last few years in the research division. Thank you.
Continuing on the topic of the plethora of research facilities in JIPMER, I would now like to invite Dr. Rajesh Nachiappa Ganesh and Dr. Medha Raj.
Good morning, everyone. I happen to be the person who kind of officer in charge of Central Research Facility, the responsibility that was given to me in the year 2019, when many clinical departments face challenges in running laboratory-based research. So this facility is essentially to provide services to departments which do not have facilities in their own department, to be able to carry out the research in lab-based work from the samples which they want to carry from their department. So this facility provides opportunities for people to come get trained and do their own research. We have 3 staff there who will help in validation and standardization of their techniques, but will not actually carry out the hands-on work, the work. And the facility actually is an institutional facility, and it provides the opportunity that they don't have to be constrained by a lack of laboratory infrastructure in their department. So it was a challenging task, and we have managed to keep it running till the year 2025, which is a surprise even for myself. We have 3 major facilities. One is a PCR and real-time PCR-based facility, which is housed in the 4th floor of this building. And we have a proteomics, which is essentially an ELISA-based facility, which is reasonably popular with most of our MD/MS dissertations. And there is a higher-level metabolomics laboratory, which is housed in this floor. The genomics laboratory, as was pointed out, has reasonably standalone facilities for all types of real-time PCR work. And we have help available to standardize and run your experiments. And so far, multiple candidates from various departments have made use of it. To our surprise, even laboratory departments do come and use this facility as a backup, because overall there is always a limitation in all the departments, and there is a backlog, and so people do come, even if the facility is available in their own department, they do come and utilize these facilities. And ELISA is another tool which is, as I had pointed out earlier, that is being used quite frequently. But actually, off late, the numbers have come down, because earlier they used to get it done mostly through biochemistry. Then there was a challenge logistically, and they were coming and getting it done here. And now I think they are able to get most of their work again back in biochemistry department. And again, a good number of users across the department. This is a far more complex instrument, gas chromatography and mass spectrometry, and high performance liquid chromatography instrument. Our GCMS has been used by at least 3 PhD candidates, whereas HPLC has not yet been put to use by anyone, but it actually needs its research use instrument. Most people prefer to use a ready-made cartridge-based straight, like you give a drop of blood and you get an answer type of result. So in addition, we have multiple facilities. The same facility is also temporarily used to help certain researchers from Department of Medicine and Department of Medical Oncology, because they have funded projects and they are not able to find space to house them immediately. So within the constraints available, we have been able to keep things running in the Central Research Facility. I think this is not a thing. We have a form. The way people can access this facility is by contacting one Dr. Raja Muthu with a PhD from our own institute, who also happens to be the technical support in the facility. He gets this form. This is essentially to create a log and find a slot so that there is no overlapping things. Time slot is given, date and time slots are given so that work is planned. And we do maintain a clear log of who uses it on which particular day, and everything gets recorded and reasonably well maintained. Many students have benefited from the facility. Thank you very much.
Continue with the Central Research Facility as part of the CRF. A bioinformatics facility was started. It made its humble beginning in September of 2023. The idea was that it should be a hub for computational biology, data analysis, and interdisciplinary research to support various research data with computational tools. So it supports fundamental clinical and translational research, analysis of various data types, and assistance to researchers, primarily for utilizing omics data from different NGS studies for translational research to enhance patient care. This is housed in the e-learning center, as has already been mentioned. It started with a bioinformatics workstation and manpower in the form of a project Saga, recruited on a contract basis, who manages the bioinformatics work. The services, primarily the NGS data analysis, including the transcriptomics, RNA-seq, miRNA-seq, whole genome and whole exome sequencing, genomics and transcriptomics, variant calling, annotation, gene expression analysis, functional annotation, proteomics and metabolomics. We made a very humble beginning, network analysis, gene regulatory networks, PPI networks, co-expression networking, and genetic model analysis. And primarily it uses the Linux-based operating system using R and Python programming. And secure data storage and management systems are available for boosting research collaborations. Custom-made bioinformatics solutions are provided on a case-to-case basis. How do we access the form? Nishad has already shown the research website. We go to the research infrastructure, scroll down the page. You have the bioinformatics facility, and there you have the link there. On clicking that form, you get this form. So the requisition form can be filled and emailed to the following email ID, that is bioinformaticsfacility@jipmer@gmail.com. And we can be contacted, Dr. Aguila and myself can be contacted with the details of the project, so that we can discuss on an individual basis as to what are the different aspects of the project and what kind of services and pipelines are required for that particular project. So far, in the last year, we have wound up 3 major projects. One from the Department of Medical Oncology, where the work was on a metagenomic pipeline. One from the Department of Dermatology, where it was the whole exome pipeline. And one from our own department, where it was an miRNA pipeline. So
So far, we have been able to set it up and made a humble beginning. The journey ahead is very important because as we move ahead, upgrading of the services are required to provide more meaningful services at JIPMER. So we will need more space. We need a high-speed internet with a server. We need more workstations. We need more terminals. Of course, more softwares. A dedicated manpower will be required in the form of a dedicated bioinformatics scientist, more bioinformaticians, a part-time IT support. This will be both for managing the bioinformatics facility as well as for conducting training from time to time. Of course, regarding research possibilities, the sky is the limit. Machine learning, mathematical modeling for therapeutic outcomes, development of databases, integrating JIPMER HIS with omics data for translational research, algorithm development for disease classification, risk management, and drug response, virtual screening and molecular docking, potential drug candidates, epigenetic data analysis for gene regulatory patterns in diseases, clinical database development for integrated genomic and patient data, development of repositories for every area of research for multi-data storage, retrieval, and transfer, web-based or standalone interface development for researchers or clinicians to store and access data, making standardized gene panels for disease, including drug metabolism for our particular population, and developing drug-gene interaction interface support system by monitoring genetic variants for specific populations. The sky is the limit. We look forward. These are exciting times to be in at JIPMER. We request all of you all to please approach us so that we can grow together towards setting up this bioinformatics facility in the times to come. Thank you so much.
Samreen, did you know that JIPMER was buzzing with this activity?
Yes, Manas, it's truly heartening. And to inspire us further, we'll now be having presentations of the winning papers in each category on stage. I call upon Dr. Bobby Zakaria, Dr. Kadambari Dharani Pragada to chair the session.
The presentation of prize winners in various categories. In this session, we will be having 9 presentations. And the first 8 presentations from the various categories, the time allowed today is only 5 minutes. So I request the speakers to stick to the time. Whereas the last presentation, best collaborative research presentation, the time allowed today is 10 minutes. So we call upon the first speaker under the undergraduate category, Dr. Abhinav Sai Krishna. Please come forward for your presentation.
Good morning to everyone who's here. Today, I'll be presenting a study which we have done on the prevalence of gustatory function abnormalities among cancer patients receiving chemotherapy, an observational study using taste strips. Introduction, this essentially 40 to 60% of patients with cancer have anorexia. This is something which we already know. Alterations caused by chemotherapy, such as dysgeusia, which is an altered perception of taste, and hypogeusia, which is a decreased perception of taste, which are seen in patients which contribute to this anorexia. Anorexia is important because it negatively impacts patients' nutritional status and the quality of life. However, there's very limited data on taste alterations in Indian patients, especially there is no data in an objective manner on how to assess the taste alterations among these patients. There are many studies which have been done in a subjective manner with questionnaires. My objectives are to assess the change in total gustatory function in cancer patients after the first cycle of chemotherapy, to assess the change in perception of 4 individual tastes. The first objective is for total taste, and the second is for individual tastes. 4 I have been testing, which are salty, sweet, bitter, and sour. And the third objective is to assess the change in the proportion of patients with hypogeusia before and after 1 cycle of chemotherapy. I will define what I mean by hypogeusia subsequently. We have done a longitudinal observational study over a period of 6 months, and the study participants are newly diagnosed cancer patients who have come to the medical oncology OPD. The inclusion criteria are all patients above the age of 18 with newly diagnosed cancer who are planned for chemotherapy and have an ECOG score of less than 2. And the exclusion criteria, there are various exclusion criteria, but overall, all of them are factors which we thought would impact the patient's taste, their ability to perform the test. The methodology, as we can see in the leftmost picture, there are 16 different strips, 4 for each of the 4 tastes, which are increasing in concentration. And those solutions are all in the 16 small containers on the rightmost picture. What we have done is we have taken a filter paper, such as the one in the middle picture. We have dipped it in each of the solutions and placed it on the patient's tongue. After placing it on a patient's tongue, they will give a response as to what taste they are experiencing. If they give a correct answer, we are giving them a score of 1. And any incorrect answer is given a score of 0. They have to choose between sweet, sour, salty, bitter, or tasteless. 121 patients were enrolled, out of which 31 were lost to follow-up. So the test was done on a total of 90 patients before and after 1 cycle of chemotherapy. Wilcoxon sign rank test was used to assess the first 2 objectives, whether the total score has changed and whether the score of each of the individual tastes has changed. And a McNema test was used to assess the proportion of patients who are hypogeusic, whether it has significantly increased after 1 cycle. Based on a previous study, which was done in a large number of normal people, hypogeusia was defined as a score of less than 9 out of 16. The baseline characteristics for the study, among the evaluable patients, the median age was 53. And as we can see, most of the patients, female patients with CA breast, which is similar to the patients which we commonly get in JIPMER's medical oncology OPD. This is the results of the Wilcoxon sign rank test. Blue is before and green is after chemotherapy. The average scores, please note that for each of the 4 categories, score is 0 to 4. So we can see that the score has decreased in all the categories. However, it has only significantly decreased for sweet and salty. However, for bitter and sour, it has not significantly decreased, and the total score has also significantly decreased. The results of the McNema test show that the number of people with hypogeusia has increased from 2 to 10, which has given us a significant p-value of 0.0. This is not part of my objectives, but something interesting to note. Please note that here in these 2 tables, I've kept one for before and one for after chemotherapy. These are the nature of the incorrect replies which were given by patients. I only want to point out one thing. which is that in the salty category, a lot of people categorizing salt as sour. Some of the limitations of this study were the small sample size and also the presence of factors, as taste is something which can be affected by numerous things. However, one of the strengths is this is one of the first studies done using this methodology, and I would want to conclude that this provides important baseline data for future studies which can use this method, and also food preferences can be determined among these patients in the future based on the nature of incorrect replies. Thank you.
For questions from the audience, I thank Abhinav for a presentation. I call upon the next speaker from the next category, that is postgraduate category. Dr. Jankala Anusha, please come forward.
Morning, everyone. Myself, Dr. Anusha Jankala, Masters in Public Health student, under guidance of Dr. Palnivel and co-authors, conducted a study titled Indoor Air Quality of Houses Located Near to Roadways in Urban Puducherry. Beginning with the introduction, almost 99% of the global population is exposed to air pollution that increases the risk of chronic disease and premature mortality. In India, air pollution is reducing by around 5 years. Indoor air quality is as important as outdoor, since humans spend nearly 90% of the time indoors. Data on outdoor air quality is currently available through different monitoring stations, whereas data on indoor air quality is limited. Hence, we conducted a study with an objective to describe the indoor air quality by measuring pollutant parameters like particulate matter, carbon dioxide, total volatile organic compounds in houses located near to roads in urban Puducherry. It's a cross-sectional descriptive study, and the study units are houses located within 100 meters, 100 to 200 meters, and 200 to 300 meters from the major roads in urban Puducherry. We have chosen a sample of 40 houses from the conveniently selected 4 clusters. And we have measured variables like distance from the road and concentration of pollution parameters. We have used a study tool called a sensor-based indoor air quality monitor, which measures particulate matter 1, particulate matter 2.5, particulate matter 10, carbon dioxide, and total volatile organic compounds. We have presented concentration of the pollution parameters as a median IQR and the line graph. And the difference in pollutant concentrations across different data is analyzed using Crystal Valleys and one-way ANOVA test. Moving on to the procedure, we have chosen 4 clusters conveniently from the urban Puducherry, besides the roads having higher traffic density like Valadaur Road, Vilupuram Road, ECR Road, and Kadalur Road. And from each cluster, we have chosen 10 houses. Then like 3 houses, each cluster, we have chosen 3 data, 4 houses from within 100 meters, 3 houses within 100 to 200 meters, and 3 houses in 200 to 300 meters. And from each house, using the air quality monitor, we have measured particulate matter, carbon dioxide, and total volatile organic compounds for 8 hours, that is from 12 o'clock to 8 p.m. on weekday and weekend. Figure 1 showing the map depicting the selected houses and clusters in the Puducherry, and figure 2 is the study tool. And moving on to the results, table 1 showing the distribution of housing characteristics and in-house exposures in the selected houses. We have measured variables like room density, type of cooking fuel, presence of chimney exhaust, and presence of trees in front of the house, pets inside the house, and number of hours per day doors and windows are kept open towards the roadway. And table 2 showing the association of pollution parameters with the distance from the road, and we have seen higher concentration of pollution parameters in the houses located within 100 meters, and there is a decrease, and this trend is decreasing, and there's a decreasing trend with the increase in distance from the road, and this difference is statistically significant with the particulate matter. Table 3 showing the association of indoor pollutant concentration, difference between the weekday and weekend, and there is a statistical difference between the weekday and weekend concentration with respect to particulate matter 2.5, 1, and 10 within the houses located within 100 meters. Table 4 showing the distribution of indoor air quality status. We have considered a house as a poor indoor air quality if any pollutant parameter in the house is crossing the WHO standard. Therefore, we have seen we have found that around 92% of the houses are having a pollutant concentration crossing the WHO standard, that is 4 indoor air quality, where it is 100% in houses located within 100 meters and 100 to 200 meters, and 58% of houses are having 4 indoor air quality located 200 to 300 meters. Figure 3 is a line graph showing the change in concentrations over a 8-hour period of time, and we can see the peak of concentration is between 5 o'clock to 8 p.m. Figure 4 is a bar graph showing the distribution of PM 2.5 concentration across the houses, and we can see the variability in the within the houses because of the different indoor sources. And figure 5 is a set of 4 line graphs showing the changing concentration across the 8-hour period in the 4 selected clusters, and we can see the peaking concentration between 5 o'clock to 8 p.m. in all the clusters except for the airport road, where the peak is in between 3 o'clock to 6 o'clock. Concluding with the discussion, the particulate matter concentration in almost all houses is above the WHO threshold, which is 15 microgram per cubic meter of air for PM 2.5 and 45 microgram per cubic meter of air for PM 10. And these concentrations are similar to the study conducted in Bangladesh. The limitation is, as indoor air quality was also influenced by seasonal variations, wind direction, and other household-related factors, which were not able to account for. Hence, we recommend indoor air quality monitoring systems and other mitigation strategies to improve the air quality of houses. Thank you.
Thank you, Dr. Anusha. Next, we'll have the senior resident, Dr. Mahadevan. Mahadevan.
Very good morning to all. Today I'll be presenting my study on the title Competency-Based Data Analysis Skill Training Program for Medical Students using Demonstrate, Observe, Assess, Perform method. So these are my mentors, Dr. Karthik Balaji and Dr. Avinash Kumar from Department of Community Medicine, JIPMER Puducherry. So background, from 2019 onwards, we have adopted the competency-based medical education for training of medical education for undergraduate students, which is shifting from a teacher-centered to a student-centered learning and an outcome-based approach. So the CBME prescribes multiple teaching learning methods to ensure the appropriate achievement of adequate skills. So the statistical analysis skill is essential for the practicing of evidence-based medicine and is a competency under the community medicine subject in National Medical Commission Council. So the CBME curriculum advocates this DOAP method for the practical skill development, but its application in statistical training is still limited, whereas this method has been effectively used in many other methods. So our objective was to assess the impact of the DOAP method on improving the MBBS students' statistical analysis skill and evaluate the competency 6.2 and 6.4 as mentioned in the NMC curriculum at the show how level through the skill assessment component. So this table shows the competency as mentioned in the NMC document and the domains and the level of assessment. So the materials and methods, the study design is like a pilot study using the Demonstrate, Observe, Assess, Perform method within a competency-based learning framework. And it included 21 third semester MBBS students in their community medicine posting service in JIPMER Puducherry. And the study tool used for assessment was a rubric for the rubric that was developed using the objective structured practical examination assessment validated by the subject expert and a semi-structured feedback that was collected through Google Forms. And also the data analysis was done by MS Excel for the quantitative analysis, descriptive statistics for the OSPI scores, and the qualitative done through manual thematic analysis of the feedbacks and reflections. So the figure 1 shows the mapping of competencies under the Miller's pyramid, which is commonly used for showing the domains of any educational tool. So here we are focusing on the shows how component for statistical analysis skill training includes like performing the data importing, cleaning, descriptive statistics, cross tabulation. So this is the flowchart of the study. The statistical training program included 2 days. On day 1, focusing on data management, software interface navigation, then cleaning, transformation, computing new variable. On day 2, on focusing on descriptive statistics, data representation using tables and figures, and research interpretation. So this happened in the e-library of the pharmacology department, where there were 21 computers provided for each of the students separately with the SPSS software. And it was implemented through this DOAP method. This was the timing that was allotted. This module was prepared, already validated with the subject expert, and was these sessions were taken in a step-by-step manner of demonstration, followed by observation closely, and they would be provided time for separate performance with assistance and later performance. So some of the challenges and solutions included were, firstly, the computer, one of the computer was not working when the solution was pairing with the nearby student. Some common mistakes were faced in the variable names, mixing measures. So all of these challenges were appropriately addressed with the solutions. On day 3, the OSPI assessment happened, where Achilles data set was given, 2 stations was prepared of 10 marks each, and the questions were projected. Each students were given individual time of 15 minutes. Later, after that, the faculty assessed and provided individual feedback, and the challenges in OSPI also was noted down. So this is the rubric for the OSPI assessment. Station 1 shows the required things that were included on day 1, and this is for 10 marks, and we can see the weightage was given to certain areas based on the consensus. And station 2 also covered the aspects that were covered on day 2 training, and more weightage was given to the interpretation. So in this OSPI assessment, there was a benchmark criteria that was picked, below which if the student fails to get the mark, they have to re-perform the station. That was 7. So the results showed that 21 students participated. The OSPI scores were indicating high performance. That was of 9.53 plus or minus 0.41 standard deviation. The mean scores in station 1 and 2 are presented. And also their post-training confidence showed a high confidence in few of the domains, that is like navigating interface, defining variables, and generating descriptive statistics. So this is the presentation of the reflections and feedback suggested. Some of the major themes emerged were the assessment process, enhancing training methods, and perceived learning experiences. So some of them were time management and having more training in SPSS, and also some positive feedback. So I would like to conclude that this DOP method for statistical training analysis showed a higher mean OSPI scores, and some of the feedback given was they had more confidence in demonstrating this basic statistical analysis concepts, and limitation included a small sample size. It was conducted only for 1 batch. So this is a effective method to teach statistical analysis skills to the students, and this can provide us a basis for revisiting the concept during their later stages of semesters. Thank you. These are my questions.
We seem to have one resident for this. Dr. Palani Raja.
Good afternoon, everyone. Our research topic was comparison of ultrasound-guided surgical clip placement and commercially available breast marker clip placement for tumor localization in patients undergoing neoadjuvant chemotherapy for breast cancer. We did this study as a non-inferiority randomized control trial. Due to advances in chemotherapeutic drug regimens, more and more patients are better tumor responses post-chemotherapy. Some patients might even have complete radiological response, meaning that the lesion may completely vanish and will become non-visualizable in ultrasound or mammogram. In such a case, it becomes crucial to localize the lesion prior to start of chemotherapy, so that post-chemotherapy there is less morbidity and patient may not require radical surgery. So, but the commercial marker clips which are available are expensive and they have limited availability. So our study was aimed at finding whether surgical ligation clip, which is routinely used in intra-abdominal surgeries, can be used as a alternative clip for commercial clip. So the commercial clip, the cost is around 5,300 rupees, whereas that of the surgical clip is around 250 rupees. And this introduced using a coaxial needle which costs around 1,400 rupees, will be comparatively cheaper compared to the commercial clip. So the primary objective of our study was assessing the ultrasound visibility of the clip post NACT, and our secondary objective was comparing the complication rate between the two groups. We did the study as a non-inferiority randomized control trial. We had randomization, and it was done as an open label study. Adult women with biopsy-proven breast cancer who were scheduled for preoperative NACT and were referred to our department for clip placement were included in our study. Those patients who were lost to follow-up or without post-NACT ultrasound and mammogram images were excluded from the study. So our sample size was 80, and excluding 6 patients who were lost to follow-up, we had 74 patients, of which 37 patients were included in the control arm and 37 patients were included in the intervention arm. Both the groups were subjected to 1 to 2 cycles of chemotherapy, following which clip placement was done, and immediate post-placement ultrasound and mammogram images were taken. Then patients were followed up for complications, and after completion of chemotherapy, repeat ultrasound and mammogram images were taken, and the data were analyzed. So ultraclip placement is a relatively simpler technique. Here, under ultrasound guidance, we infiltrate the skin with local anesthetic and then introduce the ultraclip injector needle into the core of the lesion and then press the trigger, after which the clip will get displaced into the center of the lesion. Whereas in surgical clip placement, it's slightly complex than ultraclip placement. We first give a local anesthetic, then introduce the coaxial needle into the center of the lesion, and then we remove the central sharp stylet. Then we introduce the clip through the hub of the coaxial and push it with the blunt tip into the center of the lesion. So these are the representative images showing the first image shows mammogram image mediolateral oblique view with the lesion having the ultraclip within its center. The second and third images are that of ultrasound image of ultraclip within the hypoechoic lesion and the surgical clip within the lesion. And the last image is that of a surgical clip within the lesion in mammogram. So analyzing the results, following the table showing the clinical profile, age comparatively, both the groups had similar age, mean age, which is 46 years in surgical clip group and 47 years in ultra clip group. Invasive breast carcinoma, not otherwise specified, was the most common tumor type in both the groups. And hormone receptor status, triple negative was most common in ultra clip group, and whereas triple positive was most common in surgical clip group. Response groups commonly had a partial response, which is 84% in ultra clip group and 65% in. And coming to the primary outcome, post chemotherapy visibility was 92% in surgical clip group and 89% in ultra clip group. We had calculated the confidence interval for the difference in visibility between the two groups, and it was found that the lower limit of the confidence interval was greater than the non-inferiority margin of 15%. And coming to the secondary objectives, there was no significant difference in displacement or other complication rates compared in both the groups. Then there was an added observation that irrespective of clip type, lesions with complete response, there was non-visualization of clip. This can be redressed by placing 2 or more clips together. Limitations include clip migrations in solid cystic lesions, and inter-observer variability of clip visibility assessment was not addressed in our study. So we wish to conclude that surgical marker clip can be used as a non-inferior alternative to commercial clip without compromising on clip visibility in ultrasound post-chemotherapy and without increase in complication rate. Thank you.
Now I call upon the prize winner from the category of non-student research staff, Kavya D.
Afternoon all. Today I am going to present a study, chemotherapy with low-dose immunotherapy as salvage regimen in relapsed refractory Hodgkin's lymphoma. So this is a phase 2 study. Now I am presenting the interim analysis. The coordinating centers are JIPMER and participating centers are AIMS New Delhi and Adyar Cancer Institute Chennai. So funded by ICMR. Background of this study. We know that treatment for relapsed refractory Hodgkin's lymphoma is conventional salvage chemo. With this, we'll get the overall response rate is 60 to 70% and complete response rate is 50 to 60%. But when we had the immunotherapy to conventional salvage chemo, the overall response increased 95% and complete response rate is 70 to 90%. However, the challenge is a full dose of nivolumab is very expensive, so most of the people not able to afford it. A possible solution to use low-dose immunocheckpoint inhibitors, which is 40 milligram and total dose is only 80 milligram. This is based on previous head and neck cancer pharmacokinetics data. Total nivolumab dose is only 80,000 compared to almost 6 to 7 lakh for conventional dose. Primary objectives to determine the complete response rate after salvage chemotherapy plus low-dose nivolumab in relapsed refractory Hodgkin's lymphoma. A complete response is defined as DIBL score 1 to 3 in the PET-CT done after 3 cycles. Secondary objectives, the proportion of patients who can collect stem cells and proceed to auto stem cell transplant. Event-free survival to look at the overall survival, to look at the toxicity of therapy. Study design. This is a phase 2 open label single arm multicentric interventional study. So relapsed refractory lymphoma planned for salvage chemo, a 3 cycle of salvage chemo that is an investigator choice once in the 3 weeks. These are the regimen we are using in the standard in relapsed refractory Hodgkin's lymphoma. So after completed 3 cycle, a patient underwent the PET-CT. If the PET-CT DIBL score is 1 to 3, then we will proceed for auto stem cell transplant and follow. If the DIBL score is 4 to 5, then we will consider as therapy, but further treatment is investigated choice and follow for overall survival. Inclusion, age between 5 to 65 years, relapsed refractory Hodgkin's lymphoma who had received 1 to 2 lines of prior therapy, demonstrate disease recurrence or progression. Performance status 0 to 1. Sample size, a total of 37 patients are needed in the demonstrating of complete response of 70% with 5% alpha and 80% alpha. With 10% dropout, a total sample size is 40. This is a phase 2 CMONS 2-stage design. 23 patients were needed in the stage 1. If there were 12 or few response in stage 1, then trial could be stopped for futility. Primary intervention. Nivolumab 40 milligram in 100 ml NS infused over 30 minutes before chemotherapy on cycle 1 day 1 and cycle 3 day 1. The nivolumab was used in 1 milligram of body weight. So the baseline characteristics, a median age of recruitment is 25. We have 7 patients who are in the pediatric age group. So this is one of the first trial which are looking at this low-dose nivolumab in pediatric patients. So this is some new information we will get from this study. Male patients are 77% and females are 23. First line disease is 58%, second line is 42. Completed 3 cycles of patients 85. These are the BVGV, GDP, GVDEXA, and ICE. The dose compromise 12%. ACE is 15%. The total number of enrollments is 26. After 26, we have 6 are ongoing. 2 is dropout before 3 cycles. 1 patient has died and one patient SAE. As per the protocol, we removed from the study and also patient recovered. Completed 3 cycles is 18. Out of 18, we have 17 are responded and 1 is progression. If you look at the response rate, the overall response rate is 95% and complete response rate is 67%. Partial response is 28 and progression is 5 points. Conclusion. One of the first multicentric low-dose immunotherapy trials from India. After 80 patients, we have 12 complete response. Hence, the trials continuing for full recruitment. Expected after stage 1, 23 out of 12 complete response. Actually, we achieved 18 out of 12 complete response. The regimen is well tolerated with the most AEs expected from the chemotherapy regimen. Data on transplantation and survival is maturing and will be analyzed at the later time. So no significant immuno-related AEs have been noted so far. So combining low-dose nivolumab as conventional salvage chemo. significantly will improve the overall response rate in the relapsed refractory Hodgkin's lymphoma. Thank you.
Thank you, Kavya. Next, prize winner from the category of PhD scholars. The winner is CR Akshada.
So good afternoon, everyone. My name is Akshata. I'm PhD scholar from Department of Medical Oncology. So title research is Assessment of Imaging Biomarker to Predict Pathological Complete Response in Patients with Non-Metastatic Triple-Negative Breast Cancer using Window of Opportunity Design. So this is ongoing study funded by ICMR. So triple breast cancer constitutes about 20% of the breast cancer, which is highly aggressive and which has high visceral metastasis. So a neoadjuvant chemotherapy is the standard of care in early stages of breast cancer, which ensures the early institution of chemotherapy to take care of the systemic disease, also increases the rate of breast conservation surgery, and it helps to study the chemosensitivity of the tumor. So a study endpoint is pathological complete response, which can be defined as complete absence of residual tumor in breast and axilla, so which can serve as a key surrogate endpoint for survival. So there are limited and reliable established biomarkers which are identified even before starting a standard definitive chemotherapy to predict pathological complete response. So checking the early changes or dynamic changes in the uptake value, standard value of the tumor in response to single paclitaxel and carboplatin on PET-CT scan to study the association with the pathological complete response in non-metastatic group. So this is clinical interventional prognostic biomarker study which uses window of opportunity design. Sample size is 38 and age group between 18 to 70 years who are newly diagnosed non-metastatic DNBCs with T2, T3 and T4 with or without node positive and who are planned neoadjuvant chemotherapy, especially anthracyclines and taxanes. To study the early changes in the biomarker, I have used window of opportunity design, which explores the window period. The window period can be defined as the time between the cancer diagnosis and before starting a definitive treatment. So if any intervention is given during the window period, then the study falls under the category of window of opportunity design. Once the baseline biopsy confirms triple-negative breast cancer, patient will go for baseline PET-CT scan. So if they are non-metastatic, they will receive single dose of paclitaxel and carboplatin during the window period. And again, on the 14th or 15th day, patient will go for second PET-CT scan for the assessment of SUV. So which is followed by the dose dense, which is on the routine standard of care, dose dense neoadjuvant chemotherapy, which includes 3 cycles of epirubicin and EC and 4 cycles of docetaxel every 2 weeks. So study endpoint will be assessed during the surgery, which is pathological complete response. So coming to the study findings, so 14 patients were in the group who showed the complete response and 24 patients with the residual disease. So median age was 49 in PCR and 45 in non-PCR. Most of them were premenopausal in case of PCR group and postmenopausal in case of non-PCR group. So predominant disease was T3. Coming to the nodal status, predominant was N1 in both the groups, and most of them underwent mastectomy. So around 37% of the patients showed complete response, remaining were with the residual disease. Just in the biomarker, so SUV max reduction, we have found the significant difference in the SUV max reduction from the baseline to the second PET-CT in both primary tumor and axillary lymph node. So we also compared the delta median percentage change in standard uptake value of the tumor in primary and the axillary lymph node. So only primary tumor has shown the significant reduction in both the groups. So there was a phase 2 study conducted by Connolly et al. So actually studied the significant, who have actually reported the significant reduction in the standard uptake value and correlated with the PCR So they have done it in the whole breast cancer population, but in this study, I have emphasized on the non-metastatic triple negative breast cancer. So to conclude, significant difference in the standard uptake value changes in primary tumors across PCR and non-PCR groups, suggesting the metabolic response may be associated with the pathological response in TNBCs. So I would like to acknowledge ICMR for funding this project, and I would like to thank my guide, Dr. Vishwajit Dubashi, all my co-guides, co-investigators, and my DC members for their constant support and guidance. Thank you. Thanks for the opportunity.
The next presentation is by Dr. Manu Ayan in the category Faculty Researching.
Thank you, ma'am. This is ICMR funded trial, which is actually ongoing. This is an project. This is one among the 9 top priority project which Ministry of Health and Department of Health Research is looking at to improve on selected fields in medicine. The idea behind this, or the vision behind this, is to create a integrated emergency care system in our country, which will deliver effective, equitable care to any person who gets into an emergency, thereby reducing deaths and disabilities. There are 5 centers across the country, which are distributed across to get the best capture. The rationale we look at, if you look at emergency care, as a global problem, but it's more pronounced in low and in middle income countries. And we did a country-wide assessment. This was initiated by NITI Aayog, around hundreds of facilities, and we were also part of it. We found really huge care deficits, huge problems in the way emergency care is managed, fragmented, inter-facility communications were lacking, and definitely a lot of care delays were identified, and that is the need for having such initiative. And this was also identified by WHO, and a very nice emergency care system framework was suggested by WHO and pushed towards all countries to probably implement it, basically looking at on-scene care, pre-hospital care or EMS, or the transport care and the facility level care. And in this project, we mainly aim to mitigate problems with time-sensitive emergencies. These are the 7 time-sensitive emergencies we are looking at. And the main aim is to actually develop a scalable, high-quality emergency care model, which would achieve around 80% population coverage in the selected district. So the idea is to develop a district-level model, which we are trying to assess on its feasibility, acceptability, cost, and effectiveness, and probably at the end of the thing, nationally roll it out. The methodology, it's an implementation research with a mixed method design, where we have a formative phase. We have a co-design phase where we work with the state and the district ministry, and ultimately develop a model, which is through multiple iterative cycles. The final model is the PX model, which would again be implemented across various levels of care on a hub and spoke pattern. So the provisional model, what we envisage is delivering perfect emergency care at community level by improving training, improving multiple interventions of care, improving care-seeking behavior, ensuring a pre-hospital care, which will ensure on-scene care and en route care, and then ensuring a perfect facility level care by improving of facility level interventions. We are using a CPER model, which is one among the implementation research models, which will drive our interventions. Also look at how interventions are getting effectively implemented. As any other implementation research, the District Health Authority is the implementation team. We are on the driver's seat, helping them as researchers to implement this. For our research, we have the formative research team and the implementation team and the implementation support team. and the outcome assessment team as part of the research. But ultimately, the implementers are the district health authorities. We use various methods to drive this research, where we have surveys which we do at community level. We have multiple surveys at facility level. We do specific and understand how the emergency care systems work in facilities and across different levels of care. And then the whole process started with formulating the team. Basically, all the researchers from various institutes in our district, and also the district health administration is part of the research. So till now, we have done formative research, where we have surveyed around 6,000 households in our district. We also have completed facility level assessments of all PHCs, CHCs facilities. These were across our district, how we have panned out in community surveys and health facility. The primary outcome indicator we were looking at is the adequate care, right time, at right location. And this is what we found when we surveyed around 6,000 households, that is around 20,000 people in our district. We had these emergencies noted, and trauma was one of this. And if you note, the number of deaths were really high. So skipping these, we found care deficits at facility level. We found major deficits at how patients were transported. Only 5% actually reached a hospital in an ambulance, and their spending was huge for emergencies. We did multiple facility level assessments. We did formative research by doing a focus group discussion and IDIs in patients and ambulance drivers, EMTs, and we found major care deficits. Now, what we have done till now is we have done facility mapping. This is first of its kind in our country, where we have mapped the facilities on basis of their levels, and this helps us in future designing computer-aided systems, which will ensure that patient reaches the right place at the right time. We leveraged ministry's team to actually pivot this, and we designed the first model along with the state health ministry, which contained training and capacity building at facility level and community level, also IT solution. These are part of our interventions we are doing, where we are helping state ministry to develop a call center app, which will ensure that when you call, the ambulance reaches you at the right place, and also ambulance understands where to reach, and a real-time monitoring. So where various community level engagements has been done, where we ensure that they know where to go and what to go. And there are a lot of things yet to be done. This is just 1 year past, and this is what we have done in this project. Thank you.
Thank you, Dr. Manu. That's an excellent presentation. Very challenging project, and thank you for sharing it with us. To all the best. We'll move on to the next faculty presentation of a published paper. The presenter is Dr. Mrinalini, Department of Pharmacology.
So good afternoon, all present here. I'm Dr. Mrinalini, Faculty of Pharmacology. I'm here to present the work we had done as a part of MD dissertation work of Junior Resident Arif and Hira. So the title goes as Isoniazid Monoresistant Tuberculosis Treatment Outcomes in Puducherry, a Mixed Method Study. So if we see, as you all know, that tuberculosis, and especially the drug-resistant tuberculosis, is a very it's been an existing problem. And among the key drugs, you all know that isoniazid is one of the first-line drugs. And in WHO had conducted an independent patient data analysis and found that this particular group, who are actually resistant to isoniazid, had been having an unfavorable treatment outcome if they were given a routine DSTB regimen. So in that way, they had addressed this particular group and selected a particular regimen wherein they included the levofloxacin along with the other first-line agents. And this was introduced in India as a programmatic management for this particular isoniazid monoresistant TB patients by the NTEP program in around the late half of 2018. So that is when we thought, okay, we will we will include these patients who are receiving this particular regimen, and we will have a record-based study wherein among this particular group of isoniazid resistant TB patients to describe their clinical and demographic characteristics, and also to explore the challenges, because it was a new regimen which was introduced to India. We thought we will explore the challenges in implementation of this regimen and how are the patients adherent to the regimen. We took in from two perspectives, that is from the patient's perspective as well as from the healthcare workers' perspectives. So this was a study design. So this was planned as a mixed method study. It was conducted in the duration of March 21 to December 22. And we had included patients who were started on this regimen and registered in the National TB program. And we had done it as two parts, as I had mentioned, quantitative part and a qualitative part. So as we told, like we have included all the patients who had been started on this regimen on programmatic basis of this isoniazid monocytic tuberculosis patients for the quantitative part. And for the qualitative part, we have taken three stakeholders, mainly the medical officers, the patients, and the healthcare providers who are actually delivering this regimen. And we had conducted an in-depth interview. So this provides a schema of how this study as a part of descriptive qualitative part and a retrospective quantitative part, it was conducted. So this describes that first part where when we took the quantitative part, we described the clinical and the demographic characteristics of the patients. And these were the other clinical characteristics which were noted that the HRTB regimen which was introduced had a high pill burden. As in the levofloxacin and other first-line agents which was given as a part of the regimen, this was the high pill burden was noted. And also patients had low adherence to the therapy. And with respect to the treatment outcome, which is one which is everybody is interested after implementing this regimen, was there was favorable treatment success in around 73% of them, and unfavorable treatment outcome, which included these components of loss to follow-up, treatment failure, and death, which included around 26%. And the factors which are actually contributing to this unfavorable treatment outcome was identified from the quantitative and the record-based data what we got from the National Tuberculosis Registry was low adherence, and even the culture positivity which was noted at the end of 3 months. So this you could see. And this work was just a triangulation of the work of including the quantitative part and the qualitative study, which we had done as an in-depth interview, wherein we had got the perspectives of the healthcare workers and the patients, wherein all the regimen-based issues what they had faced, the program-based and the patient-centered based with respect to their adherence, and all these factors were identified, and wherein they brought out the high pill burden, why they were actually low adherence was there, and with respect to lack of staff from the healthcare workers' point of view. So all these factors were identified, and we were able to link it. And when we see the clinical factors, the CAT gene mutation, which was more prevalent in our setting, and this was again noted, and similar findings were there with the national statistics also. And low adherence, which was seen in this particular regimen after implementing, was again been similar, and findings were there. And with respect to the unfavorable treatment outcome, wherein the positive sputum culture, which was noted at 3rd month, if it had been addressed at that particular month, the patient would have actually maybe they would have gone into a favorable outcome. That was what was brought in from the in-depth interview from the healthcare workers' perspective. And with respect to the favorable outcome, which we got from our statistics, like whatever was implemented during that time, we had a success rate of around 77%, which was contradictory to the national statistics, because they had suggested that probably Puducherry being a small state, they were able to reach to the patient and they were able to address this issue being a small union territory. So to address this issue, like we have to take care of the adherence as well as the sputum culture positivity. And this could help in formulating the other treatment programs and implementation. So this was published in Indian Journal of Tuberculosis. And I would like to acknowledge the State TB office and the team for rendering us support for the work. And I would like to also support the research division for giving us intramural research fund to carry out this work. Thank you.
Thank you, Dr. Mirilani, for your wonderful presentation. Now I call upon the winner of the category Best Collaborative Research Presentation, Dr. Murugesan from the Department of Neonatology. Dr. Murugesan, unlike the previous speakers, the time allotted for you is 10 minutes. You can make use of the time. Yeah. The other collaborator is Dr. Abhava Shastri.
Very good afternoon to everyone. I think we have had enough food for thought and we are nearing lunchtime. I realize that we need food for stomach also, so I'll keep it short. So I'm here to present our research, which is titled Clinical Profile and Risk Factors Associated with Elizabeth Kingi Anopheles Infection. This was conducted as a case control study as part of an outbreak investigation. The collaborating departments were ours, Neonatology and Microbiology. So I would like to draw your attention to this ballpark number, 10 million. So 10 million is nothing but the number of new deaths we are going to face every year, directly or indirectly related to antimicrobial resistance. And in fact, 3 years ago, we audited our own unit data with the antibiogram and saw that 55% of all the infections were due to drug-resistant organisms. So why this is important is because we are discussing about a multi-drug resistant pathogen which is emerging. This is known to cause infections in vulnerable preterm infants. And the problem is they are otherwise stable. They don't have the traditional risk factors like indwelling catheters, lines, or tubes, and it causes meningitis. So it's an important factor in morbidity. So that's why we are about to discuss about this pathogen. And this is Dr. Elizabeth King, after whom this organism has been named. So I'll tell a brief history of what happened in our unit. So in 2019, there were 2 cases of this novel organism, but the species was Elizabeth Kingia meningoseptica. So we did a quick environmental surveillance, and it was found that the organism was growing in tap water and one of the breast pumps also. So we did some literature search and found out that after cleaning the overhead tanks, chlorination, and improving the protocols for cleaning the breast pumps, we were able to control the infection. But we were wrong. It only returned with greater vengeance, should we say, in the form of a new species. This time it was Elizabeth Kingia anopheles, and it was protracted. The problem was this time we couldn't find anything in the surveillance cultures, and we reached a point where this was the leading organism causing sepsis in our NIC. So we thought we had figured it out, but probably no. So we had to go back to the drawing board to find out if we are missing something. So we decided to answer our questions with a research question. So our research question was, what are the risk factors for Elizabeth Kingia anopheles infection compared to those who get other bloodstream infections? Because I told you this particular subset, they don't have the usual risk factors for infection. So the primary objectives were to identify the risk factors, and the secondary objectives were to analyze the clinical outcomes. So this was an exploratory matched pair case control study conducted in our unit. So we started the study in 2022. We got waiver of consent. So cases were those symptomatic neonates who had infection with Elizabeth Kingia anopheles, and controls were those who had infection with other bloodstream infections or other pathogens. So we did matching for gestational age category, time of NICU admission to ensure that the cases and controls have been in the NICU for the same period of exposure, and onset of infection, because we know that early the onset, the infection is usually from perinatal sources and mother. Later onset is usually hospital acquired. So we decided to match for that also. So we made a list of exposures which are possible after a lot of brainstorming. So these were what we could come up with, the causal pathway right from delivery of a baby to what will happen when the baby is in the NICU. So maternal risk factors, delivery room interventions, and factors in the NICU. So we made a list of all these and decided to collect these exposures from the case sheets. So some of them are highlighted in red because we couldn't collect them as it was a retrospective study. So to identify all the exposures related to this infection with an odds ratio of 5, we needed a sample size of at least 38 cases and 38 controls for a power of 80%. So we used standard statistical tests and also followed it with a conditional forward binary multiple logistic regression model to find out the adjusted odds ratio for the significant exposures. So this was the study flow. We were able to get 42 cases in that time period, out of which we had to exclude 4 because some were polymicrobial, some were identified as contaminants, and we decided not to treat them. And one was asymptomatic. So we were left with 38 pairs of matched cases and controls. So coming to the baseline, I would just draw your attention to the similarities. They were comparable, but the important thing is 3/4 of them were preterm with an average gestational age of around 34 weeks and a weight of just above 1.5 kg. So these are the exposures at the time of onset of infection. You can see that those in group 1 or case 1 cases, they have a relative stability at admission. They didn't require any resuscitation. Half of them didn't require even any respiratory support. So they were more likely to be only on enteral feeds rather than IV or parenteral. So they were likely to be cared in the less sicker areas of the NICU, like level 1 or level 2. And they were more likely to be cared by mother rather than the nurses or the relatives. So even symptomatology wise, the other control group, they manifested with severe symptoms like shock, whereas the case group manifested with milder symptoms like apnea. So there was a distinct phenotype which we could recognize when we were even treating these babies, but this proved our findings. So when we put all these pieces together and did a logistic analysis, we found 3 factors were significant. Those who were only on feeds, those who were relatively stable at admission, those who were likely to be cared in level 1 or level 2 of the NICU were more likely to get this infection. So we thought the first one was more relevant because there is some collinearity between the second and third category with the first one, meaning that the second and third category of infants are the ones who are more likely to be on total enteral feeds. So this brought us to the question, is there a breach in feeding equipment sterilization? So no new exposures we could identify in this study. And the worrying thing was 76% of all these isolates were multi-drug resistant. They were only sensitive to levofloxacin and minocycline. Even morbidity wise, the control group or the other BSIs, they were having more incidents of shock and death, whereas this case group had more incidents of meningitis, ventriculitis, and hydrocephalus. That means it was a more morbid infection in a group which is less likely to have infection otherwise. So there was some concern in published literature about this organism being mistaken for Pseudomonas. So we did a Sanger sequencing of 10 representative isolates, which confirmed the sequence identity of this organism. So we did a few interventions. By the time we finished the study, it was much later, but based on what we already knew, we decided to act immediately. So some of these interventions, which we would say were major, was we decided to autoclave all the feeding vessels. So these are our feeding vessels called pallidi, and so we decided to autoclave the feeding vessels or the pallidi, which are made of steel. And education about how to clean the breast pumps, we revised the protocols and all. No other major protocol was change was made during that period, except for some minor changes in feeding protocol, which we don't think would have contributed much. And this is the time trends of the infection timeline. We can see that it's a 4-year data from 2021 to 2024. We can see that this infection was rampant in 2021. In fact, there were months when there were 9 cases of this infection. And we made this intervention somewhere around April, May 2022. And it took us a month or 2 to streamline these interventions, after which we could see a dramatic decline in the number of incident cases. So we still have some infections occasionally, but there is no clustering or no propagation of this epidemic. And at least in the last months, we have had no infections. So coming to the discussion, this is an environmental pathogen, initially thought to be relatively harmless, but it's gaining its virulence because of antimicrobial resistance. So it's supposed to be there in water, taps, contaminated milks, and flush solutions. And one thing which is worrying is source identification is often difficult. And even if we do environmental decontamination, it is proving to be ineffective. So which was what happened in the second round of epidemic which we had. And shifts on housekeeping and infection prevention methods. Of course, we couldn't collect all exposures because it was a retrospective case record-based study. And we didn't know about what happened to those babies who are getting discharged from our NICU and going and getting admitted elsewhere. So we are probably in some sort of a canary in a coal mine situation, because canary birds are usually the ones which coal mine workers take to their caves, because they are more sensitive to poisonous gases and carbon monoxide. So coal workers know if canaries are getting sick, we are the next to get affected. So the question we should be asking ourselves is, antimicrobial resistance in neonates is really a sign of things to come in the future. So we feel that infection control is the urgent need of the hour. So have we reached the end game? Probably no, because infections are still the #1 cause of morbidity and mortality in our setting. And this was a research driven more by need or urgency to improve clinical outcomes. So it would be incomplete if I didn't thank those involved. especially the ground level people like our residents and nurses, the housekeeping staff, who helped us implement these practice changes. And HICC team especially, because they are always just a phone call away, and their blood culture turnaround times are probably one of the best in the country. So their support is very invaluable to us. And of course, our own department faculty, because we have had a lot of discussions regarding this, and brainstorming was done, and faculty project. And that time I was just fresh from residency. So, but I received adequate support and mentoring from the rest of the faculty, so which was very remarkable. So I would thank all of them, and also thank you all for your patient listening. Thank you very much.
Thank you, Dr. Murugesan. And that brings us to the end of this session, and congratulate all the prize winners across all categories for their excellent work which they have shared with us. Thank you very much.
Congratulations to all the prize winners. Your presentations were fantastic. I thank the chairpersons for steering the session.
Now we'll be breaking for lunch.
Yes, please announcement for all the students, lunch has been arranged in the dining hall onto the left side. And for all the faculty, dignitaries, committee members, volunteers, please go to the MPH hall first floor. And please try to be back by 2 p.m. Thank you.
Request everybody to be back by 2 o'clock. We have more exciting sessions in the afternoon also. So request everybody to quickly finish your lunch and join back by 2 p.m.
Up next, we have the faculty award presentations in basic sciences and clinical or nursing sciences. It is a privilege to call on stage the chairpersons for this session, Dr. Bhavna Bade, officiating director, and Dr. D.M. Thapa, Dean Research.
Welcome and very good afternoon all. We'll have now JIPMER Research Award in this, and I request Dr. Rajesh Nachap Ganesan from Department of Pathology to have his presentation. He has received the award in the morning.
Thank you, chairpersons and the audience for this opportunity and the honor. It's quite a rare thing for a surgical pathologist to be awarded for research. And because the branch that we are in, we are constantly engaged in service work. We hardly find much time for research, but thanks to the MBBS course, we get categorized with hardcore research department. So we are kind of a misfit either way. And the service work, again, doesn't give much room to fancy about and to maintain energy, motivation, and to get funds to do further research. It's not an easy task in the branch that I am in. So in spite of all those things, it's a great honor that the Institute decided to honor my work, because there is nothing too hi-fi in the research work that we do. My research and practice started in 2002. It was kind of an accidental thing. Two close relatives passed away due to renal disease in the same year. I was in my residency period. And incidentally, my guide was a flamboyant person. He left to Sydney on a foreign sabbatical for more than 2 years. And he had committed to help certain PhD students who were actively engaged in renal research. And that burden, he said, do you mind helping them out? So that was the compulsion that came into me. Those days, we never had the courage to tell no to our guide. That's the system that we, at least I had to come from. But I had no way to know anything much about renal pathology, and their work was very hardcore. So I had to go to multiple centers to try and learn. And that's how the whole thing started. My own dissertation work was on ovarian tumors. It was funded by Lady Tata Memorial Trust. It was on phosphatase and tensin analog and mutation in ovarian tumors. And some part of the work we had published here and there. And my original first paper on my own work came in the year 2007, much later after the work was actually been done. But the work that came out of those supporting my other PhD candidates came in much earlier. This was one paper which I will never be able to forget, because this endothelial nitric oxide synthase is quite popular nowadays, but that was first time demonstrated in tissue in India by our own team in experimental models, because before that it was a hypothetical concept and serum levels were determined, but tissue demonstration was never done. This was done in juxtaglomerular apparatus in kidneys. And most of these kits were all mostly by donation from United States. We just did not have the infrastructure. We, of course, had the infrastructure. We kept on modifying with whatever little tools that we had. Most of these were innovations, and the kits were all mostly by borrowing from, like after they tried, they failed several times. So they had some remnants. We agreed to spare the shipping fee, and we got these reagents and tried in our setup, and it worked. So several other papers came in with multiple collaborators from many countries. So I'm just trying to show a few papers which were not in any way connected with my own course or my own, dissertation. All these were entirely in an aspiration to help, which I had promised. And to do that, I had to take a lot of help from Christian Medical College, Vellore, whom I will never be able to forget. It's not that they had great deal of expertise. There were two GI pathologists. One was Dr. Susie Kurian and Dr. Anna Pulimud. Most of these studies also involved a hectic amount of ultrastructural work. And they both were actually GI pathologists. When I said that I need help, they said we will all learn together. So that's the way we did most of these work. And subsequently, I could get benefit of training from IKDRC Ahmedabad and PGH Chandigarh, Dr. Kusum Joshi and Aruna Vanikar. But my initial thing, most of these things were, most of the work in renal research happened primarily for those PhD candidates whom I had promised to help. So that's how my beginning into that area was. After working in several other institutions, I landed up in JIPMER in the year 2010 and had a chance to start the renal pathology lab. It was a great struggle. It was not easy, but still the service is continuing, and we have made it into a routine functional service without any major disruption. Of course, we still do not have the electron microscopy facility, but for that we do most of the other work. I always had a feeling like I never got a certificate for my renal pathology thing, because in my time, there was no formal course for a fellowship. These fellowships were actually like through International Society of Nephrology. Not that much learning happened through these courses, but essentially these are just certificates from some internationally reputed organization. But there was a chance to do, and I also got these certificates. I have so far guided about 5 MD students, and my first independent student came in the year 2014. And then the next thing happened. I actually wanted to become a PhD candidate and do research myself as a candidate. But somehow I was jinxed. I could never find a guide who was willing to take me. So there was a rule which I found out that you can be your own guide, provided you are eligible to be a guide. So I decided to take a PhD guideship. But it so happened that Professor Parija was, I think, the Dean Research or Director, I'm not sure. The moment the guideship came, he also ensured that all of us are allotted candidates. I somehow didn't have the courage to say no, and I had to take up that candidate. And we also started the postdoctoral fellowship program in nephropathology in our department. And I also was made this associate dean research, which was again a very humbling experience because in my own department, I did not fit the profile of such high designations, but was given this responsibility. And this PhD candidate that I had to take made me kind of, literally I could say, forced me to apply for multiple extramural projects to ensure that sufficient funds are available. Because the candidates have come, there is a responsibility and we have to do justice to the candidate. And after several failed attempts, I did manage to get my first independent project from DST SERB. And after the funds came, I realized I just didn't have the lab space. Now I have to buy an instrument, I have to have facilities to store reagents and multiple other stuff. And I will always be indebted to Professor Siddharaju because he actually shared his room. The room that was allotted to Sir, we exchanged. Mine, I was a junior faculty, so I had a smaller room. So we both mutually exchanged. It was a great gesture. And there was some other space in the department which was not used. They were mostly condemned areas. We made every single possible available area and made use of it to continue with the project. And so that journey kept continuing. I eventually ended up getting 3 PhD candidates so far whom I have guided. And the second PhD candidate, by this time I have got more experience. I also got additional funding, this time from a different agency, from Department of Biotechnology. And this was the second PhD candidate. And at the moment, I have an ongoing project in molecular oncology. As a pathologist, definitely the oncology-related services constitute the bulk of our work. And we keep doing this renal thing as an additional part of the specialization. And I had a chance to, actually I had an invitation to go to United States, but the institute authorities said that unless you have government-funded fellowships, you are not permitted to get salary. I was very clear, I'm not going anywhere unless the leave is with salary. And thanks to COVID, there was DST, DBT, and ICMR all had closed down any foreign-related funding. And this was the only option that was available. I successfully got this funding. This also ensured that I got my first visiting professorship. I thought I'll go there as a trainee, but I don't know on what the basis. They had their own regulatory committees and they said that you will be a visiting professor in our institution. And it was a great honor to be in that institute in the United States. During the same period, I also had a chance to do some certificate course in molecular oncology from Thomas Jefferson University. And with that, I will just highlight a few areas of research work that I have done in the last few years. I know it can be a bit hectic if it is for a non-expert, but I'll try and keep it as simple as possible. This was a work on graft biopsies. We get to see some thrombi, microthrombi in fine, tiny capillaries. And those we generally attribute it to hyperacute rejection, or whenever we see a microthrombi, we are worried about the long-term outcome of these transplants. So not much details are available. And so far, the largest series of such graft microthrombi in post-reperfusion biopsies were 8. That's the largest series that is available from all over the world. And I had the chance to study more than 1,500 renal biopsies in the same settings, and we could get that was a huge number for this particular condition. And these were the thrombi. These were antiplatelet CD61A antibodies, just to highlight that they are in the glomeruli and in the peritubular capillaries, etc. We also did ultrastructure to distinguish these microthrombi from other non-specific structures which can mimic microthrombi, particularly tubular epithelial cell fragments and other things which cannot be distinguished by light microscopic finding. So the point why we were interested is we wanted to know what happens to these grafts when you follow them up, when they, those patients who had these thrombi, what happens to them when you follow them up for much longer. We could ensure that we had a median follow-up of close to 7 years. And we found that those who had diffuse microthrombi, ended up having a higher risk of graft failure. And they also had a stronger association with circulating panel reactive antibodies. This essentially meant that there is an antibody-mediated insult to these grafts, and they are not just a bystander. That got published in a reasonably reputed journal. Kidney International is one of the highest impactful journal in the field of nephrology, and they agreed to accept this article because of the rarity of the content and the completion of the workup that was done. Then the second study, which was again published recently, was vancomycin nephrotoxicity. Vancomycin casts, one vancomycin is used as a last step drug in multi-drug resistant bacterial infections or sepsis. Most of the time in intensive care unit, patients admitted in intensive care unit with sepsis and multiple other injuries. So acute kidney injury, we cannot attribute it whether it is due to the drug or it is due to the multiple comorbidities that the patients are suffering from. And a major problem is we cannot also devise an animal model to study these. On the other hand, patients are also not biopsied very commonly. Patients are so sick that renal biopsies are not performed in such settings. Generally, when we have such conditions, when we want to study and understand the disease biology, we tend to do an animal model study. But here, animal models are not feasible because you cannot induce such a severe disease in an animal and try and do an animal model study. And just administering vancomycin per se will not reflect the clinical situation what the patients are in. So that was the context in which we studied again. It's a very rare condition. I don't know what is happening to the slide. The slide is not moving. We studied about, we had to review the records of close to 5,500 patients over an 11-year period. And identified at least 50 patients who had administration of systemic administration of vancomycin and an episode of acute kidney injury, and also had biopsies because we want to find the changes in the kidney biopsy. That was our primary aim. The main intention is trying to distinguish those who truly have vancomycin nephrotoxicity from those who do not have. Because once you suspect vancomycin nephrotoxicity, you are going to withdraw the drug. And withdrawing one of the last line drugs without sufficient evidence may also be counterproductive in a patient with multiple infections. So that's the setting. And finally, we had to have a very strict inclusion and exclusion criteria and ended up with 36 patients who had this vancomycin nephrotoxicity. The challenge is lots of data had to be collected. Of course, it was a Herculean task. Like it was a very extensive work of the serum levels, the time temporal association between the administration and the acute kidney injury, and a whole lot of other work was done. And we also developed this antibody, which was not available in the, it's not, commercially available antibody. This is a vancomycin immunohistochemistry directed against the vancomycin cast. This was done to actually validate that these are truly vancomycin casts. They were, we attempted to distinguish them from the other casts, which can be, mimickers of vancomycin casts, like uromodelin cast, myoglobulin, etc. And we also did ultrastructure to identify the structural characteristics of these vancomycin casts and attempted to distinguish them. From this study, we could find certain characteristics which were reasonably helpful in distinguishing the features of vancomycin nephrotoxicity from the group that did not have. So essentially, this was a very rare kind of a study which I had to take up, and I had to read a lot of pharmacodynamics and pharmacokinetics of the drug, at least for this study, because it was not easy, and the journal kept asking us to extensively rewrite the discussion part on the pharmacodynamics aspect. We had our strength. Most of our co-authors were either pathologists or intensive care physicians. They, like, none of us were having the expertise, but all of us pitched in together and worked on that article and could publish it in a reasonably good journal. And already there are a reasonable number of citations on that article. And this was another study which we had done recently, where we had used studied the reproducibility of biopsy features. and try to compare the human pathologists. We had blinded 4 renal pathologists and attempted to score few parameters that we normally do. Already aware that we are going to be abysmally poor when it comes to concordance, because we generally never agree with each other. That's a human tendency, and more the expertise and more the experience, we also tend to differ more. So that's we were already aware. What we expected was when we use artificial intelligence tools, probably our concordance rates would become closer. That was the aim. And as a part of that, we had to create a lot of cartoon models. The challenge that is, though anyone who is practicing in the field will be aware that all these are like very well spelled out entities. But when we wanted to do a research study, we realized that most of these are our personal understandings and there are no clearly spelled out definitions. So to create these definitions, we had to write to several experts or those experts who had already made some definitions and get some consensus definitions and created some cartoons so that among the 4 renal pathologists, there are no discordant views. And it so happened that in United States, most of the consultants were also from different countries. One expert was from, of course, they are all United States citizens now. One was from Vietnam, the other one was from Spain, the third one was from Italy, and the fourth one was a German person. I think one was an Egyptian too. We had people from multiple different backgrounds, so their training background is also different and probably their understanding was also different. So we had to clearly define these areas, and we had used 5 different AI tools. These are like open source, pre-validated tools for pathology related work. We had to do this machine learning thing. Anyway, I always have had an interest in IT, this thing. So for the first time, I did have a chance to work on my own coding and started auto annotation for areas of interstitial fibrosis. 5 different, like 4 renal pathologists and 5 artificial intelligence tools. So these were the various hi-fi looking images. But when we did the concordance with the usual kappa score that we do, between the human pathologists, the concordance rate was somewhere between 0.5 to 0.74, which was even higher than what we expected. We were much more happier with the numbers that we had, because that's absolutely normal. But when we compare the different scores among AI, they were not even, they just don't agree. They don't talk to each other and they don't agree on anything. And we try to correlate it with the estimated GFR on follow-up. The patients are followed up for close to 5 years. I think 4.8 years was the median follow-up. And we try to see what the human scores could predict at the end of 5 years. Most of these scores had a reasonably good prediction, irrespective of which pathologist, what score was used. Most of them are quite significant in predicting the renal decline over a period of 4 to 5 years. Whereas they were not at all significant, irrespective of which method we had used. These methods were not significant for the AI tool. So it was a shock to us, but we decided to, it was a negative result kind of a study because we anticipated the results to be completely on the opposite side. But what we learned from this was that the artificial intelligence and machine learning tools can be extremely precise and they're very, very robust in quantitation. But they have very poor reproducibility between systems. And image, we use these slide scanners for whole slide imaging. And what is scanned with one software, you cannot extrapolate and feed it into another software because they just don't accept that image. Then you use a third-party software to modify and somehow make that image be read. So you'll have to do lots of management-related issues and multiple software-related aspects were there. We could handle most of that, but what we could learn was AI tools can be useful adjuncts. And probably if we all use the same software and the same tool as a guiding tool, probably it will be able to give us a quantitative measure for the same team of people. So it's not as easy or as rosy for AI tools and to be in direct application in practice soon. This was another paper I'm I'll try and keep it simple. There is this entity called polyomavirus nephropathy, which happens mostly in immunosuppressed patients in renal transplant settings. And this BANF is a group which keeps giving scoring systems. The most latest followed scoring system was very complex. It so happened that one of the leaders for that group was a person who had invited me originally to United States, because actually I criticized him on a meeting in India that the scheme that you have devised appears to be too complex and won't be practical. So we had come up with we tried to validate, but I had also come up with a devised model. And what we found was that the revised model, or the new index that I had proposed, had much higher correlation with the plasma levels of PK virus and had much better correlation in terms of the outcome. It so happened that finally I had to send this paper to the same person, to a journal where he was the editor. And he was more than happy to evaluate the whole thing, did the scrutiny very thoroughly, and accepted the work.
Dr. Rajesh, you have exceeded your
Thank you.
time.
This was a work done by one of my PhD scholars, but the bench work, some part of the work, I had to do it myself, because by the time this work had come to its completion, he had lost steam. He wanted to complete the course somehow. And I had to do this work myself and found that the TNFR2 levels had significant outcome-related association in predicting the behavior of the disease in terms of relapse and progression to end-stage renal disease. We had to do this levels in the serum, in urine, gene expression, as well as in tissue, and prove that this is quite a robust marker. To complete, the major challenges that we face are we hardly get any clinical correlation in our setting. And there is no pathology interpretation without clinical correlation, and that's the background in which we work, and our research is also quite skewed. And ours is mostly a one-way service. And future direction of my research, I want to keep it as open as possible and keep exploring and let the opportunities decide and keep diversifying more and more. I would like to thank a whole lot of people. I have not named anyone specifically. My family, my teachers and students, my departmental colleagues, and all the technical staff without whom we cannot do any of the work. Ours is a large department with over 150 staff members, and each one contributes extensively. Since this presentation is essentially on renal research, I would like to mention the departments of nephrology and pediatrics who do contribute significantly. But I will always mention departments of biochemistry, microbiology, and radiodiagnosis, because our diagnostic workup can never be made without correlating with these findings. And I would say all the medical and surgical departments, because essentially coming from abroad, we work closely with everyone. And I have continued to, I started my journey with collaborations outside, and I continue to collaborate with multiple people, both within and outside the institute, and I would like to keep remembering them always. Thank you very much.
Please give a big hand. So, Dr. Rajesh, heartiest congratulations, and bringing pride to our department also. Thanks for that, and wish you all the best for your future research.
Thank you.
The next awardee for the JIPMER Research Award in Clinical Sciences is Dr. Adisivam D. from Neonatology Department. Let him make his presentation.
Respected Director, Dean, beloved teachers, colleagues, students, and my pathological friend Rajesh. Good afternoon. So, the universe is vast. There are millions of stars. Some are twinkling, some are blinking. But whatever you say, every particle in the universe matters. Every particle will conspire something better, right? Welcome to our neonatal universe. We have a lot of stars distributed among 3 levels and also the postnatal wards. Of course, we work in tandem with our neighbor,
the obstetrics department. So in the next few minutes, I'll give you a glimpse of what's happening in the Department of Neonatology, part of research and clinical care. Mind you, whatever happens, whatever betterment happens with clinical care, research improves and vice versa. So they are actually inseparable, at least in the context of neonatology. Breastfeeding is the fundamental thing for life, right? So I believe most of you are sensitized for the August first week where we celebrate the World Breastfeeding Week. But we do continue to promote breastfeeding all 365 days. So this picture is part of the Bonding Angel program. By the time Lakshya program was initiated, we had already started the Bonding Angel program. We had our labor room, which was always busy. The turnover was high, nurses were busy, and sometimes there was not enough hands to take care of the babies to the breast of the mothers, right? So, but we had the nursing students there. So they, most of them were trying to help what was going on, but a majority of them were passive watchers in the whole scenario. So what we did was we started labeling them as the bonding angel of the day, gave them badges. Their only job was to take the baby to the breast of the mother so that early breastfeeding is initiated. It worked out well. Mother was happy, baby was happy, sisters were happy, and the nursing students also received the certificates. They continue to receive the certificates every year. I thank the nursing college and the nursing team for this wonderful innovative. And as I said earlier, though breastfeeding is a physiological thing, especially for primary mothers, someone had to talk regarding the important hair issues and some postnatal lactation support had to be given. So if it was done at the right time, much earlier, there was no problem at all, right? So our first extramural grant was on postnatal counseling using video, right? We, for the first time, we had a lactational counselor in JIPMER because of a project. A dedicated nurse was appointed only for talking on the postnatal mothers regarding breastfeeding and making sure taking postnatal lactation support. It worked well. So after that, we also had few publications related to breastfeeding. During COVID epidemic, my colleague, Dr. Tanigainathan, along with him, we did a project because there was a lot of fear that whether the COVID virus is transmitted through breast milk. And in fact, during the first wave, many babies, despite the mother and baby were stable, they were separated. So that led to a lot of problems. So we could, we did PCR in the milk samples, oropharyngeal swabs for the mother as well as for the baby. We could document that none of the breast milk samples transmitted the COVID virus. So we were happy that during an epidemic, we had scientific proof that breastfeeding was doing good. And this has been done by one of our MD dissertations, an RCT which was done for post-LSCS mothers. Why post-LSCS mothers is quite simple. When we talk of early initiation of breastfeeding, for spontaneous vaginal delivery, most of the time it was happening, say 15 minutes, 20 minutes, etc. But when it went to post-LSCS, sometimes it took hours. Surgery was done in the same hospital, same block. Mother was in one cubicle, baby was in another cubicle. So there were a lot of barriers, artificial barriers. So to circumvent that, at least for in the sake, in the cover, in the sake of research, we wanted to improve the or bring down the time for babies for the first who were born post LSCS. So for that, we used an RCT using domperidone in one arm and placebo in the other arm. Whether it worked pharmacologically or as a placebo, we found definitely effect with the domperidone arm. But basically, it was the add-on lactation support and postnatal counseling which was done. Then as an extension of our breastfeeding activities, we started the Human Breast Milk Bank. As an undergraduate, this was a quiz question for us. Do you know whether a human milk bank exists? We were really happy that we could initiate in 2016. Now it's almost 8 years, thanks to the wonderful work of all the nursing team behind the milk bank and the lactational counselors who are doing it in the administration for their wonderful support. So after initiating it, we were able to supplement for the time period. It's not a permanent solution. For those babies who had transient insufficient milk supply, we are able to substitute from the milk bank. The same thing which happens in Ponley, that happens in a large scale. If it's a wet grinder, what we are having is a mixie. It's a small effort, but we are doing the pasteurization. Post pasteurization, the milk is doesn't contain any microbes, and after testing it microbiologically, then we are feeding the babies, right? So when we started it, we also had a research component. Thanks to Professor Vishnu Bhatt, he guided me through my part-time PhD work. We also did some research aspects of human milk banking. So we did a pre and post study. We found that there was a decrease in the neonatal mortality, necrotizing enterocolitis. So in fact, human milk banking is not a competitor for exclusive breastfeeding. Wherever a human milk bank is established, it has a ripple effect in the community, and exclusive breastfeeding actually improved. That's what we found. And we did an RCT with the fortification and also the Holder pasteurization regarding the lab components, namely the protein, fat, the macronutrients, and Ig and IgG, which actually was found to be decreased. Having said that, though pasteurization decreases the nutrient composition of breast milk, the immunological composition of breast milk, it is 1,000 times better than the formula milk. Moving on to the next area, major chunk is neonatal sepsis. Right from term babies to preterm babies, neonatal sepsis is devastating. Dr. Murugesan was also pointing regarding Elizabeth Kingia. So, in the context of neonatal sepsis, we were interested in the prevention. ICMR devised a probiotic study by giving a probiotic to low birth weight infants, whether we are able to prevent sepsis. That was a research question. So it was happening in 6 sites. We were one of the sites, and the results are being analyzed. For this study, we were really happy that our efforts were not confined to the walls of NICU. We went to the community, right? In the community, those babies, those low birth weight babies who received either the probiotic or the placebo were followed up by the nurses. Imagine for 2 months, a dedicated nurse paid home visits, more than 16 visits for that baby within 2 months, which an A&M in the routine program also would not have done. So by that way, we were able to take care of more than 700 babies in the trial over a period which crossed over rains, COVID, a lot of things happened in between, but we are able to complete the trial thanks to all the study physicians and the field investigators. Next is a NEO-OP study. Dr. Nishad is also a co-PA for that study. This is also an international study which was done in almost 11 countries and 19 sites. And it's an important study wherein they have found out the spectrum of neonatal pathogens and the antimicrobial resistance associated. This is in fact is the first phase wherein they have identified these are the organisms causing the major problem and this is the antimicrobial sensitivity, but done at a systematic way across countries. So this was out of this study, a lot of papers have come out. Out of important ones are the global burden of antimicrobial resistance in 2019. This was published in Lancet. So actually, JIPMER, we have put it in the global map because of this NEO-OP study. Then we had the patterns of antibiotic use, pathogens, and predicting mortality. NEO-OP score was also developed. Dr. Murugesan has spoken in length about Elizabeth Kingia. So this is a gram-negative rod and has caused havoc in outbreaks. And we were fortunate to have one undergraduate student who did a G.J. Strauss project with us, and it was also published. Then we had a couple of PhD scholars who worked with us. The first scholar worked with late onset neonatal sepsis, that is sepsis which occurs after 72 hours. We had done some basic work, global DNA hypomethylation. She found that there was indeed a significant DNA hypomethylation associated, and also with the expression in neonates with late onset sepsis. There also she found that the expression was reduced. The next important topic is therapeutic hypothermia. Conventionally, most of the babies who do not breathe at birth have a brain injury. So this is birth asphyxia. So for that, most of the centers actually don't cool. But the concept of cooling came initially, they started with selective head cooling. Then it moved on to whole body cooling. In the whole of India, it was pioneered first in CMC. Apart from CMC, the center which has most studies in therapeutic hypothermia is JIPMER. Thanks to Dr. Butt and his constant pursuing the area of therapeutic hypothermia. So in therapeutic hypothermia, we keep the baby's temperature between 33 to 34 degrees Celsius, then rewarm the baby. 72 hours it's being done. So these are some of the recent studies which are published in the area of therapeutic hypothermia. One of our DM candidates, Chanchal, did a study whether therapeutic hypothermia effect improves along with, suppose I add Maxelf and it has got neuroprotective effect. Suppose I do both therapeutic hypothermia and Maxelf together, does it improve? We didn't find much significance because we need a much larger sample size after that. Then we have also published regarding long-term outcomes of therapeutic hypothermia and neuronal biomarkers in predicting neurodevelopmental outcome in babies with asphyxia. The second PhD scholar had an MPH background. So he, Dr. Dinesh, he did work on the risk factors for adverse neonatal outcomes. So he was trying to read on the maternal risk factors which are known, and from that to predict the adverse neonatal outcome. So in fact, he developed an app called Janam Map, right? Janam Map, JIPMER antenatal risk factors for ANM. That's the short form. But of course, this is in the primitive phase, needs to be extrapolated. Then comes neonatal transport, whether it's extramural or intramural. Intrauterine transport is the best way of transporting babies. When it comes to neonatal transport, we have seen that most of the babies, even though 108 system is better in South India, some of the newborn babies, though using 108, are also reaching hospitals with hypothermia, basically because they're not stabilized before shifting. So we developed a sick neonate score for babies who are being transported. So that has been validated subsequently in the other studies. And for intramural babies, whether we can prevent hypothermia at admission, that is also being explored currently. Next is kangaroo mother care. This is a wonderful thing, kangaroo mother care, right? All good things in the world come in small packages without much advertisement. KMC is one, right? So putting the baby skin to skin contact and exclusive breastfeeding does wonders. So in this area of KMC, we have seen whether KMC reduces the anxiety among mothers. We also tried to introduce music plus KMC therapy, right? And other QA activities are also being devised for KMC. I will be short of my talk if I don't mention regarding QA. We have got QA champions in my team, right? So for almost every important aspect, be it outbreak or sepsis, improving breastfeeding, or improving the donor milk output in the human milk bank, a lot of QA activities was done. Dr. Sindhu Sivanandan was also here. She also did some good QA work. In fact, we should appreciate OG team for getting certified in Lakshya, and we are also in the pathway of Muskan. So this is just an example. One of our DM candidates did a QA activity to reduce the breastfeeding, the time from the delivery till the first feed. That's called DEL FBF, first breastfeed time. So to start with, it was more than 60 minutes, right? After that, she did some PDSA cycles, 1, 2, 3, 4, 5. All right, lot of PDSA cycles. Each one I cannot elaborate, but subsequently she has brought it to 20 or 20 minutes, right? So that's a wonderful thing, and even in being sustained for a longer duration. So this is the milk donation QA which was done along with the nursing staff. There also we could, because of simple QA activities, we can improve the breast milk collection. Our important partner in neonatal care in research is the OG team. That's given a greater share for them. And these are our minor partners, ICMR, Bill Gates, they're all our minor partners. So having said this, out of so much research, etc., so what is the proof of the pudding? The good news is that you see from Jan 2014, right? It's almost a decade data here, which is before you. This is NMR, neonatal mortality rate, right? Per 1,000 live births. So that has been hovering around 40 to 50 in, say, 2014. And it has come down to around 10 in 2024, right? It's a great thing. Then the bloodstream infections for 1,000 live births, which was also in 70 and 80 for 1,000 live births, has come down to now around 10. Despite all the Elizabeth King outbreaks, we have been able to achieve this. So thanks to the entire team of all stakeholders and our neighbors. So I will be wrong if I don't mention regarding the nurses. Nurses are the backbone of our NICU, right? So they have been taking care of our babies 24/7 for into 365 days with all their activities and also supporting us with all the QA activities. Some of the nursing students also do their research with us. Even though being short or brief researchers, they have provided wonderful inputs for us. We also do training, constant training for nurses as well as for doctors, especially on research station. That was our Amaju Singh, our alumni training on cranial ultrasonography. This is our own alumni training on ventilation. We also participate in public awareness campaigns, breastfeeding, sepsis prevention, etc. And thanks to the nursing team and the nursing college for supporting us for a long period. So every gardener knows the joy of seeing the first sprout. It's a symbol of hope and endless possibilities that lie ahead. So that's true. And our nursery is no exception. There are brilliant minds sitting before me and a great future awaits. So thanks to the main stakeholder, our main star, the baby, the VIP. So he is the who entrusts their babies into us, into our building, and all the stakeholders, the entire team members of NICU family and departments. We have collaborated with almost all the departments. Probably geriatrics we would not have collaborated. Otherwise, we would have collaborated with all the departments. So thanks to all the stakeholders, we wish to take it forward. Thank you for the wonderful opportunity.
Thank you, Adesyavam, telling us the various baby steps which we can also undertake to give care to our babies. So and highlighting the various research work done in neonatology by you and your predecessors have got the help. Thank you very much. With this, we close this session. We move to the other session then.
Thank you, sir and ma'am. That was an informative session from which all of us can learn so much. Moving on to the presentations by the Department of Anatomy, Clinical Pharmacology, and Medicine. Please join me in welcoming our esteemed chairpersons for the next session, Dr. Vikram Kate, Dean Academic, and Dr. Lata Chaturvedra, Professor of Obstetrics and Gynecology.
Good afternoon, everyone. Again, happy Women's Day for all the women sitting here and the supporting persons who are sitting here. Thank you, sir, for asking me to start the session, and thank you, organizers, for giving me this opportunity. Without further delay, we'll move on to the department presentations. For this, first we have Department of Anatomy, and Dr. Sarsu, the HOD, will be presenting.
Thank you, ma'am. Madam Director, Dean Academic, Dean Research, my senior and junior colleague, and dear students who are eagerly waiting for the day to get over. A very good afternoon. And let's see what we can do in research in Department of Anatomy. At the outset, I would like to thank for the opportunity. It's an honor to represent my department and a great opportunity to walk you through the research activities in the Department of Anatomy. So before I get into the core topic, the research, I just wanted to show you the graphics, anatomy graphics, what we have and what are the core areas, how the spectrum of anatomy has spanned into four key domains. Okay, so we have one, the academics, two, the patient care, and three, the research, and four, the administration. So these are the key domain of our spectrum of work. Under that, academics forms the major key component of our work. In that, we have at least 13 courses with MBBS, nursing 2 batches, and MLT and AMS, total strength of 420. At PG level, apart from MD, we do have MSCs, physio, biochemistry, and neurotech. And do we have other certificate courses also? So together, it's around 495 students per year. We deal with that in teaching and learning activities. So next component comes, this is a vital area where much of our time is, substantial time and energy is spent for fostering this academic excellence. The next is the patient care. The patient care, we do have a lab called cytogenetics. It sprouted like a research lab. Later on, with the permission, expertise, license, etc., we converted that into a patient care. Though research activity still happens in there, it's more of a patient care service right now. Here, what we do is we examine the whole chromosome. It's the whole genomic chromosomes level study. It's not the gene study, it's a chromosome study. Okay. And then we do have research activities, projects involving faculty, students, PhDs, etc. And our department plays a very key role in assisting the administrations. Many of our faculties are in a key positions as associate dean and faculty in charges, section in charges, committee chairman, and secretary. This also takes much of our time and energy too. So going on to the core research area where it is a topic of interest today, let me take you through what are the facilities available in our department and what are the expertise we have to handle that facilities, and later on progress to the research activities. Okay, under the facilities, we can categorize into different kind of labs. We have the lab, cytogenetic lab, which functions both as a patient care as well as research, where we do chromosomal studies. Any kind of structural or numerical aberrations in chromosome above 5 million base pairs. Below that, we will not be able to pick up in them. Okay? A chromosome defect more than 5 million base pairs. And if it is a gene level studies, SNP studies, or expression studies, or association studies, we do have a molecular genetics lab where we do the initial part of the DNA extraction, and later on collaborate with other departments for further study, like real-time PCR or anything else. But we do have a basic facility for that. And we do have this microscopy lab where we have high-end fluorescent microscopy with the confocal facilities, and which can go in for in-depth dimensional study where out of focus is not there, 3D dimensional. We can take serial sections and construct an image, and it's a high-end microscopy. And we do have tissue processing, where when you get a tissue, it converts itself into slide. That process of tissue to the slide happens in the tissue processing lab. And it is also a core area for research. And we do have a cadaveric lab. I would say it's a cadaveric lab because much of our research do happen in this lab where we have cadaveric studies, etc. So these are the facilities. And a few equipments, main equipments, I would say, what is available in this area. This is the cytogenic lab. This is the fluorescent microscopy. You can see this, and this is a system which is like more than 30 years old. Right now, we are trying to get it updated to automated system. So you can see it is a whole chromosome. The 23 sets of chromosomes are there. So what we can do with this, we can see for any structural or numerical aberrations. where we can see structural and numerical aberrations, like, for example, our collaborating departments are obstetric gynecology, pediatrics, endocrinology, even medicine. So cases like primary amenorrhea, infertility, and a lot many cases from ambiguous genitalia, developmental delay, all those come, and we just take the peripheral blood and send it for processing and look for any aberrations. Okay, so that's about the chromosomal aberrations, if it's structural or numerical. In molecular, what we do have all the small equipments. One equipment which is high-end, which has been shared with other departments, is this gel doc. We take the blood, do the DNA extractions, and after DNA extractions, you send it through a gel and put some electrical component and run it, and this is how it looks like. So the gel, what you put it inside, is being captured like a picture by this equipment. And this has been shared by other departments also. So this part of it. After this, we collaborate with other departments for further studies. So this is what your DNA is. The DNA has moved, and this is the ladder through which you can compare where depending on the size, etc., you can compare your DNA. So this is what this equipment does. This is a gel doc. The microscopy lab does have the fluorescent. This is the fluorescent microscope, and it's a specialized fluorescent microscope called the confocal microscope. It is like a pinhole where all the images concentrate to that. It's a mechanism. We can do many things in this. This is how it looks like. There's a wide field fluorescent microscope and a confocal, where different, just like a CT scan, you can take serial section image and you can get the depth and width of that. So this, but right now we are using this for immunohistochemistry, but many things can go in this, can happen in this fluorescent microscopy. The tissue processing, we have all the high-end tissue processing equipment. Right now we are equipped with that. So any tissue you get it, we can get to the slide format. After that, you can proceed on to whatever you want, the staining procedures or something like that. This is our tissue processing. The cadaveric lab, I cannot say much of the equipment. We do have a deep freezer where we can freeze everything. And then later on, we get it into the processing. So this has been this cadaveric lab. Much of the workshops also happen here in this cadaveric lab. And we do conduct workshops in karyotyping, immunohistochemistry, and everything. So this is the research facility and equipment available in our department. And we do have expertise to operate these equipments, and to have a patient care of genetic origin, we need to have a certain expertise. Unless you're trained, you will not be able to give the report. So a few of our faculty have undergone a training, special training for this. So this is a genetic diagnostic 6-month fellowship at CDFD. It's a DBT funded fellowship by Government of India. So after this, actually I have gone for this fellowship, and we are trying to establish the diagnostic, not just karyotyping. Karyotyping is one extreme, sequencing is the other extreme. In between, we have pockets of small diagnostic tests where we can address many things in between these two. And one more faculty, Dr. Dharmaraj, he has undergone a clinical embryology course, and right now he is assisting in the IV lab for doing the embryology process, a clinical embryology process. So these are the a few, apart from this, small trainings our faculty undergo for respective research area of interest. So based on these facilities and training, we do have a lot of projects. These are the projects which has happened only in the past 5 years, not prior to that. Why I'm insisting 5 years? Because in 5 years, we had a pocket of COVID where everything was at baseline. So before that, we did have many projects. So in this 5 years, we had some 14 faculty and MD projects and 1 PhD and 2 other co-investigators and co-guide. And some non-funded projects like cadaveric projects, etc., also happen. Some medical education projects also happen here. Right now, some 8 projects are ongoing, and 1 PhD is still there doing this. In the field of research is our labs are the field of research. Most of it is in cadaveric because this was a COVID time, and most of the study was in the cadaveric, followed by our genetic study and some immunology. We do collaborate with radiology department and other departments for other type of studies. Right. So based on the studies, we did have a research. These are the past 5 years performance. You can see this is the area of COVID. And prior to that, we did have a lot, or maybe we did have more of case reports and medical education. Now we started to do a wholesome projects. Both are the reasons why our publication is slightly on the lower side, but of course, now everything is in place. We will be picking up the research output from here. And we do, apart from the research publications, we do have scientific presentations. This yellow bar is the scientific presentations. As we started to become, as a senior faculty, the course faculty, people started to call for more of a course faculty and, guest lectures. So this is what the green has increased significantly higher because our faculty go for more of guest lectures and course faculties. Yes, we do have significant amount of scientific presentations also. And with this, the facility available, we have managed to conduct few workshops. In 2019, we have conducted a karyotyping workshop. It's a national workshop. Later on, the COVID set in and we were not able to do it. In department initiated workshops are just a little bit coming down. We will be picking up too. What you can see is the collaborative workshop, like cadaveric workshop and other workshops have picked up. Many departments collaborate as a notified workshop. Apart from this notified workshop, we do have internal training where cardiology, surgery, many residents come and train us. That is not a declared workshop, it's an internal training. Every year when we do thorax, we'll call the cardiology. When we do neuro, we'll call this. Those training also happens apart from this declared workshops. So these are few things which happen in our department. Apart from this, our faculty have significantly contributed in publishing books, many books, and have been an editor of Grace. These are single-authored books, and co-authored many books and written some chapters also. These are significant amount of time goes for, if you want to sit and write a book, it's not simple. It's a big work. So one of our faculty will be presenting. So he has authored many books and it's a great effort from his side. So with this background, we have some basics and we are in the progress of taking our labs to the higher level. We do have some 5-year plan. So we have planned that immediately what we can fix and upgrade is like our karyotype, which is like little 25-year-old to make it into automated and introduce some fish, which is not just a patient care, but also a research component where we can expand our territory to amniotic fluid and etc. That is the immediate future plan. So we are expecting we will be finishing this by next year and there's 90% chances of getting it done. In 2026, once this is ready, we don't want to go to the sequencing part. In between, we have tests like multiplex ligand probe amplification, TP-PCR, triplet prime PCR. These are something which is cost effective, just like a fish. We can detect deletions and duplications and triplet repeat disorders in projects and etc. Here, it is comparatively low cost and low manpower, etc., compared to the sequencing part. So this is something like people don't, explore this. So we are trying to establish this. It requires only a thermocycler and a capillary electrophoresis. And yes, manpower is little required. And with these two components, I think we'll be able to establish this. Anything with deletion duplication, for example, DMD or spinal muscular atrophy, where many too many genes are deleted, and in one go we can do the estimation. So that's the thing. And next is the cell culture lab. We have started the process. We have started a project. One of our faculty has started, and we have just started the cell culture. To establish and to make it fully functional, it will take some time. So by 2027, I think we will be establishing a full-fledged cell culture lab. Later, if possible, if all this in place and we have enough of manpower, etc., we are planning for the same capillary electrophoresis we can use for whole exome sequencing or targeted exome sequencing. This is the plan, and it will open doors for many research and patient care services. And of course, cadaveric lab and long-term plan is a plastination lab that is also in the pipeline. It in another 1 or 2 years, maybe we will get a software for students' point of view, which can be used by clinicians also. That is in pipeline. Already we are working on it. we might get a full-fledged cadaveric lab. Maybe this will happen much earlier to all the other things also. So we are expecting that to happen. So this is our future plan, and we are just working, all of us are working together to make this happen. Hopefully, when I come back, if at all someone comes back after 5 years, they will be updating whether we are successful in getting this. This is our research update, which has happened. We have some 23 lakh intramural fund and few extramural fund, which I think it's missed in the slide. So mostly in the external, we have to improve on the extramural funding part also. I agree to that. Moving on to some relevant project, which we just completed. I will start with simple project like a prevalence, which we have done in a cytogenetic lab. Then we will go into a molecular cytogenetic. Then we will take another project where we have involved the confocal microscopy. So this is just a simple project done in our lab, just to see the prevalence of chromosome abnormality in primary infertility. It can be done in recurrent pregnancy losses. It can be done in primary amenorrhea, etc. So this is just to say what is the abnormalities here. It was a 2-year study with 192 participants, and the blood sample was taken. Karyotyping banding is done, and we see this. This is how, this is a few samples, few observations of the entire thing. So you can see this. You can see this is what is this? Students, if you're awake, what is this? What is this? Okay, Klinefelter. Yeah, this you may not be able to see. This is the 9 inversion which has happened in the P, and these are people who are infertile, come for infertility. The karyotyping and genetic is the last resort they go to. They exhaust all type of investigation, etc. And if nothing is happening, they come to the karyotyping. I would suggest that they have to come. They can start off with the karyotyping as simple as that. You can see this is the mosaic done, ma'am. And this is 18 and ring here, and this is deletion. I'm done. Okay, because one more is there. So mostly this is the structural and so this is one male who had come with XX. So basically his genotype was X, but he's a male, established male, and he came with infertility. So this is the thing. So infertility they have to work on. This is a small thing we could do. Can you present one more?
You're already time up. If another minute you can finish.
Yes, yes. Two other from cytogenetics, little molecular cytogenetics, only application.
So, good afternoon, and I'll take a minute to explain this. So this is one of our PhD project which we have taken up, and the title is Cytogenetic and Molecular Characterization of Teenage and Young Adult Lymphoblastic Leukemia. So this is a collaborative project together with medical oncology, pathology, pharmacology, and you could look at the karyotyping which we have done in our Department of Anatomy, and this slide which is being shown there. I'll go to the outcome of the study. The outcome is we have come out with a low cost effective method to identify PH like ALL in these patients, that is teenage and young adult patients. So this is a collaborative project. So I would, and these are 2 publications which have been come out of the project. So I would like to thank medical oncology, pharmacology, and pathology for their support. And thank you.
So this is another study that we are carrying using the immunohistochemistry and confocal microscopy on anterior cruciate ligament in collaboration with the orthopedics department. Usually we say that anterior cruciate ligament is avascular structures, but by using the biomarkers in immunohistochemistry, we have seen that it is a vascular structures. And rather than removing it, right now we are planning for the cell culture lab where we will effect of plasma on the ACL cell cultures. So we will see the effects of the platelet-rich plasma in the cell culture of anterior cruciate ligament. So, and hope so in the future we can go up to the skin culture for the patient care also in the cell culture lab. These are the future plans. Thank you so much.
Thank you for the lucid presentation from the Department of Anatomy. And we know we work in close quarters. I hope pre-implantation genetic diagnosis and prenatal diagnosis really takes up soon. Thank you, Sarasu.
Thank you once again, Department of Anatomy, and now invite HOD from the Department of Clinical Pharmacology, Dr. Sandhya, to present her the departmental work.
A very good afternoon to one and all. First of all, I would like to thank Director and Dean Research for giving me this opportunity to present about Department of Clinical Pharmacology in 10th Research Day. So in the next 20 minutes, I will be giving an overview of the background of the department, the research facilities available, funded projects in the department, our top 10 publications, and the research impact of our publication. So to begin with the history of the department, in 2009, the DM Clinical Pharmacology course was started under Division of Clinical Pharmacology by the former professor and head Department of Pharmacology, Dr. C. Adityan. So I joined the second batch, that is in 2010 as a DM candidate. So in 2013, July, the Department of Clinical Pharmacology became independent from Division of Clinical Pharmacology from Department of Pharmacology with Dr. C. Adityan as head. And in 2014, January, I joined as assistant professor in Department of Clinical Pharmacology. Dr. Adityan retired in 2015, and I took over as head of the department in 2021. And during this period, Professor Ravindran, Dr. Jayanti, and Dr. Manigandan from the Department of Pharmacology took care of the department as officer in charges. And in the meantime, we also started PhD program, and currently 2 PhDs are pursuing PhD in the department. And in 2021 December, our department got selected as one of the advanced centers for clinical trials under ICMR, Indian Clinical Trial and Education Network intern program. In addition to the regular teaching, we have Adverse Drug Reaction Monitoring Center functioning under Pharmacovigilance Program of India, through which we collect ideas from various clinical departments and report it to Indian Pharmacopoeia Commission. We also have a Drug Information Center, which provides evidence-based drug information. And we promote ethics and good clinical practice in research by conducting annual GCP and bioethics workshop. And most of these workshops are conducted in association with Division of Research, JIPMER. Coming to the facilities available for research in our department, we have got a cardiopulmonary exercise test apparatus with bicycle ergometer to measure exercise capacity or the peak oxygen consumption, VO2. And it also has the facility to do pulmonary function test. We also have a cardiovascular analysis system to measure pulse wave velocity and arterial stiffness, a ambulatory blood pressure apparatus, hand dynamometer, and other lab-related equipments like ELISA reader, washer, cooling centrifuge, deep freezers, pharmaceutical refrigerators, and biosafety cabinet. So looking at the research done using these facilities, from 2014 till now, these are the number of tests or the number of tests done for thesis purpose using CPET, the body box plethysmograph, DLCO, 24-hour ambulatory BP measurement, pulmonary function test, etc. And in addition to these facilities, our department also provides support in conducting randomized double-blinded placebo-controlled trials. So if you look at these drugs available here, there are various oral formulations, capsules, tablets of different colors, drugs in different blisters, syrups, powders in sachets. So if you look at both drug and placebo, they look alike. So we talked to the pharmaceutical industry and tried to procure similar looking placebo for the collaborating departments and help them in doing randomized double-blinded placebo-controlled trial. So coming to the research projects going on in the department. So the extramural research projects with our department as the primary investigator is, as I already mentioned, ours is one of the advanced center for clinical trials under Indian Clinical Trial and Education Network, INTENT by ICMR. So this INTENT is a program to promote clinical trials across academic institutes. And whenever ICMR wants to do some of the important clinical trials, it will call for expression of interest for intent centers, and only the centers under intent can apply expression of interest and do the trial. And the budget is given on a approved project basis. So as and when a particular institute takes up a project, the budget will be released for that. So currently we are pursuing a project under ICMR intent, and it also happens to be a MedTech Mitra ICMR CDSU initiative project to find out the effect of a chemically treated filter paper as a blood sample collection tool for measuring serum concentration of first-line anti-tubercular drugs. And we are also currently part of a IGIB initiated study to find out the genetic landscape of COVID-19 across India. So we are one of the 10 sites. Now coming to the other extramural projects, which is going on with other departments. Some of the prestigious projects are like Center for Advanced Research and Network of Oncology Clinical Trials with Medical Oncology, and Efficacy, Safety and Tolerability of the Ayurveda Regimen with Dr. Sonali Sarkar. And we also have a study in OG, where initially it was a multi-centric study, but later converted into a single center study with respect to JIPMER. It is initiated by JIPMER, by Dr. Nivedtaja. As the other site was not able to recruit patients and do the randomization as per the protocol. Now it has become a single center. So these are other collaborative projects going on as extramural projects in the department. The last two being ICMR faculty project, ICMR PhD project carried out by our faculty from dermatology and ENT. Okay, now coming to the intramural projects. So these are all the intramural projects where I have guided the students. It's currently ongoing, and most of our studies are randomized controlled trials, and it will be revolving around either vitamin D or curcuma longa. And these are the ongoing projects as co-guide. So most of the collaborative projects where I am a co-guide is either because that's a randomized control trial where we have to provide the support of randomizing and getting placebo for the study, or because of the CPET, exercise capacity, to find out the exercise capacity, or to do causality assessment with respect to adverse drug reaction. So that's the pattern of research which is going on in our department. Either it's a RCT or finding out the exercise capacity in various conditions, or related to adverse drug reactions. So these are the extramural projects which were completed, and some of the sponsored clinical trial where I was a co-investigator, which got completed. So again, this was my intramural project, faculty project, which got completed, and thanks to JIPMER for the intramural given to my faculty project. So these are all the completed projects as co-guide. So as I already mentioned, most of these projects are related to RCTs. So coming to the details of most impactful research projects in the last 5 years, some of the things I would like to share, like this is one of the study where we compared 3 different scales to do causality assessment of drug-induced liver injury. And following this, we got an opportunity to write the following article, hepatitis B vaccine and immunoglobulin, because of this causality assessment in drug-induced liver injury. And this is a initiative taken by our SR during one of the journal club presentations, when we thought, why don't we compare 3 scales and find out the causality. So a simple journal club discussion turned out into a publication, which again brought us an invited article. And again, these are all some of the case reports published from our department like azinocomaral as an alternative anticoagulant in patient with warfarin-induced nephropathy, a complex interaction between anti-tubercular treatment and azinocomaral. This was a patient who was on azinocomaral and was going to be started on ATT for tuberculosis. And so we were measuring, so we were following up the patient to see what kind of interaction she's going on, how the dose should be adjusted, etc. And this is one, the third one is again an interesting study where we came out with a novel desensitization protocol for infliximab hypersensitivity in a 13-year-old patient with pustular psoriasis. The patient was having hypersensitivity for infliximab, but they can't afford for another drug as the patient is from a poor family. So we went out for this novel desensitization program. So these 3 case reports, which we got because of our ADR monitoring center and drug information center, resulted in we getting one more publication from some other center, that is Kolkata Tropical School of Medicine as well as Arjikar Hospital, Kolkata. The investigators and the clinical pharmacologists from there approached us for writing an article on 10 clinical pharmacological interventions in routine care to ensure better treatment outcomes. So of that, 3 of the case reports were from our department. So they included this and co-author for the same. However, today I would like to present one of the studies which was carried out by my PhD scholar and which got the E. Mehlingam Award in this research day, titled Molecular Docking of Curcumin and Curcuminoids as Human Zinc-Dependent Histone Deacetylase Enzyme Inhibitors. So this is a part of her thesis. Actually, her thesis is a RCT, randomized control trial, to find out the effect of curcuma longa and cholecalciferol in DLBCL. So this one part of the study. She also has a cell culture work. So this was published last year. So the objectives of this study were to virtually screen curcumin and curcuminoids. That is D-methoxycurcumin and bis D-methoxycurcumin, again human histone deacetylase enzymes in comparison to their pan-HDAC inhibition activity with FDA-approved human HDAC inhibitors. So I would like to give a brief introduction about this study. The curcuma longa, which is the turmeric, it has got 2 components, curcumin and curcuminoids. The curcuminoids are D-methoxycurcumin and bis D-methoxycurcumin. So most of the studies are carried out with curcumin because curcumin bioavailability is better as compared to other curcuminoids or curcuma longa as a whole. So if we look at literature, most of the studies are with curcumin. And curcumin has been found to have this pan-inhibitory action. So we thought, if curcumin is having, what about curcuminoids, which is also a component of curcuma longa, whether it has got pan-HDAC inhibitory activity or not. That's what we wanted to study, and that is our objective. And we also wanted to predict the drug likeness property, absorption, drug metabolism, excretion, and toxicity of curcumin and curcuminoids as compared to already available HDAC inhibitors. So coming to the rationale of this study, HDAC enzymes catalyze deacetylation and they cause silencing of genes, resulting in downregulation of tumor suppressor genes. So HDAC inhibition has been found to be a potential tumor target, and there are already many FDA-approved HDAC inhibitors are available for treatment in the market, but they have got their own adverse effects. And as I already mentioned, there are many studies which states curcumin is a pan-HDAC inhibitor. Our study wanted to find out whether curcuminoids are as effective as curcumin in inhibiting HDAC enzymes. So if you look at the work, it was a molecular docking work. So through literature from PubChem, curcumin and curcuminoids, that is DMC and BDMC ligands, as well as the already available FDA-approved HDAC inhibitors in the market, chidamide, panabinostat, romidepsin, etc., were retrieved from PubChem. Similarly, HDAC enzymes 2, 3, 4, 6, 7, 8 were already available and they were retrieved from protein block database. However, those HDAC which were not available readily were retrieved by homology modeling. And these, we found out whether these ligands are going and binding to this HDAC enzyme. And it was validated using Autodac Vena software as well as Ramachandran plot. And the results were analyzed. So what was the result? So if you look at the overall docking score, that is the affinity with which curcumin and curcuminoids bind to the HDAC enzymes, shows that the curcuminoids, that is BDMC and DMC, they were having their docking score almost similar to or slightly more than that of the already available HDAC inhibitors in the market. For BDMC, the docking score was highest as compared to curcumin and DMC with minus 7.1, which was even similar to or more than that of romidepsin, SAHA, trichostatin, etc. And other thing which we checked was like whether this curcumin and curcuminoids have drug-like property. For that, we have to check whether they follow Lipinski's rule of 5. So Lipinski's rule of 5 states that to be a drug molecule, to be considered as a drug molecule, the compound should have a molecular weight of less than 500. The number of H-bond donors should be less than or equal to 5. Number of H-bond acceptors should be less than or equal to 10. Lipophilicity as expressed with log P should be less than 5, and molar refractivity should be between 40 to 130. So curcumin has got all the properties to be considered as a drug molecule as it adheres to Lipinski's rule of 5. So from this, we found that among the curcuminoids, this bis-D-methoxycurcumin has better permeability, and both curcumin and curcuminoids have a reasonable molecular weight, permeability, lipophilicity to be developed as a drug molecule in future. Even romidepsin, which is one of the drug molecule available, HDAC inhibitor available in the market, has got a higher molecular weight with challenging oral bioavailability. So to conclude, this study found that curcumin and curcuminoids interact actively at the binding pocket of HDAC enzymes, and they are potent HDAC inhibitors as compared to the already available HDAC inhibitors in the market. They follow the rule of Lipinski's rule of 5. Hence, they can be developed as a drug. And based on some of the software, it is all ADME software, it is also found that they do not cross the blood-brain barrier. So their toxicity and mutagenic property are also less. So if they are developed as a drug in future, they may be better than most of the HDAC inhibitors in the market. But however, this is a molecular docking work. So these findings need to be proven further with animal studies as well as human studies. So coming to the top 10 publications as per impact factor. So these are all the research work published from our student thesis or faculty project. And all the top 10 which I have included is with our department as the primary author or corresponding author. And the work done in our department in collaboration with other departments. So the impact factor starts from 6.1 until 1.1 in the top 10 order. So almost all the equipments and the facilities available in the department, we are trying to use for research, and we have published, we are taking care to publish almost all the works. So these are some of the works published with cardiopulmonary exercise testing apparatus by us as well as collaborating departments. And this is the work published from our body box plethysmograph, which was doing DLCO, pulmonary function test, and from our drug information center and ADR monitoring center publications. So these are all pharmacovigilance related publications at various time points, our case reports. And in addition, so all the work done in the department as thesis is also presented in various conferences, and some of the prestigious awards are this UK Sheth Award for Best Oral Presentation and Dr. A.D. Joseph Award, Best All-Round Presenter from South Asian chapter of American College of Clinical Pharmacology. And in two subsequent years, two of our DM residents got this award. And Dr. Saibal Das, one of the DM candidate who is currently working in ICMR, during his tenure, he wrote an article to IGME in a competition related to bioethics and got winner of Maiden IGME Young Bioethics Award. And Saibal also, when we wrote one meta-analysis on vitamin D in migraine, he got a travel grant to go to Dublin to present this work. And again, Saibal Das, this ISRPTCon, Indian Society for Rational Pharmacotherapeutics, When they conducted the conference in JIPMER during 2014, they came out with one JIPMER medal, and he happened to get that JIPMER medal when he went and presented it in Kolkata. And coming to these awards and prizes, these are all by my PhD scholar, where she presented her work in various conferences. And to add to that, today she has got the E-Mahilangam Award for one of the work done by her. So to summarize the research activities done in the department from 2013 till now. So these are the ongoing and completed intramural and extramural projects in our department. So blue in completed and pink in ongoing. So in intramural, with our department as a primary department, 8 projects and 2 extramural projects are going on. Well, these are the intramural and extramural projects going on with collaborative departments. And in these 12 years, we have published nearly 88 articles, including original, systematic review, meta-analysis, etc. So, I would like to thank our previous head and all the officer in charges of the department, administration, director, dean, academics, dean, research, and MS for providing all the support, the collaborating departments, my students and office staff, without whom this work will not be able to do, because ours is a small department. So ours is a relatively small department and a new department with a 12-year-old history. And we have started our research and we are going slowly and steadily towards our goal of achieving a clinical trial unit in future to conduct phase 1 clinical trials. So we are moving forward. With this, I would like to say thank you.
Thank you very much, Dr. Sandhya. I will not use the word small department. It's a wonderful department, emerging department, and you have achieved quite a lot in a short span of time. And right now you are recognized for the clinical trials also in the country. That's a big achievement. And you are giving good support to everybody who are actually doing clinical trials. So once again, I would like to congratulate you as well as all the people who are involved in the development of this department. Started within the Department of Pharmacology, then it's separated, and now a student of the department. I'm happy to see that she's heading the department. Thank you very much. Thank you for your presentation.
Thank you, sir.
Congratulations once again, Sandhya. Now we'll move on to the last but not the least, Department of Medicine. And I welcome Dr. Vivekanandam to talk about his department.
Good afternoon, respected Director is not there, Madam is there. Good afternoon. Good afternoon, the Dean Academic, Kate, Dean Research, and his big team who is organizing. And the chairpersons also. I'll present the research activities of Department of Medicine on behalf of our department. Happy to be in front of you. So, of course, day to day we are involved in huge clinical load. Our main areas are infectious diseases, tropical conditions, and non-communicable diseases. We have, of course, not much of research facility in the sense of equipment, etc. Of course, we have a centrifuge and a freezer that amounts to our equipment. We are largely clinical research, so we have a team of highly dedicated and skilled. We have a faculty mixed with various interests and various skill sets. So it's quite fascinating to work as a team. Because this is a research facility. This represents the research facility, and of course, our residents taken a get together. So these are the main research facility we have. And to put our department in the context, I think it is not often well understood. We have around 280 to 300 patients under medicine. That is about 1/6 of all IP. Actually, 1/2 of all patients on ventilator at JIPMER are under medicine. I think it's not understood that way. Half the ventilated patients are under medicine. And 1/2 of all deaths that happen in JIPMER happen under medicine. So that tells you the volume and the complexity and the illness severity of our patients. It also gives us opportunity of a wide variety, and also we are not to certain aspect of medicine. We admit all medical emergencies. The casualty doesn't have to worry which it belongs to. Is it neuro, nephro, gastro? They don't have to worry. They just give to medicine all medical emergencies. We are happy that way. So that is our background. I am mainly presenting data for the last 5 years. Of course, 5 years was a memorable beginning 5 years ago. The first case of COVID in India was January 20. The last 5 years, initially it was COVID. The government of India created a COVID vaccine trial support unit at various places. JIPMER also was one of the sites, and Dr. Kadiravan coordinated that. And that platform trial unit helped evaluate 2 vaccines. Okay, so Covovax is a nanoparticle vaccine. Of course, these are all multi-center industry-sponsored trials. We also contributed. This was subsequently approved by CDSO based on this trial. It's a nanoparticle vaccine utilizing a spike protein coated on nanoparticles. And then another vaccine was a first mRNA vaccine from India, that was a self-amplifying mRNA vaccine, which is a novel concept. This trial also we were part of a multi-center trial, and Dr. Mukta was the site PI for this. And also we got enrolled for the landmark recovery trial, the trial for COVID, evaluating various options, and conducted by the Oxford University. Then, of course, another multi-center study in COVID was about secondary infections in hospitalized COVID-19 patients. Again, ICMR multi-center study. Dr. Venkatasubramanian was the PI here. And all sites, it is about 18,000 patients. And the secondary infection rate was around 4%. Overall, COVID mortality 11, but those with secondary infection had a mortality of 57%, underscoring the role of secondary infections in mortality. And the pathogens were 90% bacterial, some fungal, a significant proportion of polymicrobial infection. So these are the main pathogens, mainly Klebsiella and other gram-negative. And there was a significant AMR, like Acinetobacter was 90% carbapenem resistant, Klebsiella was 70% carbapenem resistant. Then another important area for our research is the snake bite. And of course, there are about 4 important studies. There are several others. I'm not highlighting all studies. One is evaluation of a oral drug, very interesting, Varisplodib. Then another is an infrared thermal imaging to identify envenomation. Another is a severity score, venoms. Another is a outcome measurement set development. Varisplodib is a phospholipase A2 inhibitor. Phospholipase A2 is a significant component of the venom responsible for various manifestations, and oral Varisplodib has been developed as a inhibitor. The Bravo trial, again, it's a multinational study. We were part of that. Of course, Varisplodib did not make a difference in the primary outcome, which is improvement at 6 hours. But there was a secondary outcome in those who received the drug early. So probably early administration is probably crucial. And there is a follow-up study, Varisplodib as an IV drug for 6-hour infusion, that is just has been completed. So probably it will come as a important molecule. Let's hope. This is an orphan area, so anything is welcome. These are the secondary outcome was different in the sense of AUC day 7 was different. The IV is going on. And the second study on the snake bite was, can we use a thermal imaging? Will it help in identifying local envenomation? So in this study, the previous study, Dr. Chanavirappa was the PI, and here 3 of our faculty, including Chanavirappa. This, like if you do a thermal imaging, the bitten part, if there is local envenomation, it looks warm, hot. So there is a hot spot can be identified by thermal imaging. Compared to this, you can see this is much more red and white. So that means higher temperature. It was evaluated, and it was found that the infrared imaging has a sensitivity of 94, which is very good, but specificity is somewhat limited at 83. Maybe it could be a tool in the periphery when there is a clinical uncertainty. It needs further evaluation, but it's an interesting idea. Of course, simple and low-cost device. Another, based on our patients, about 200 patients, a mortality prediction score was developed. This is completely a JIPMER study based on our patients. And this is by one of the postgraduate, Dr. Maya, along with Dr. Chanavirpa. A simple mortality risk prediction score or Viper in India, Vinam study. There was a model development as well as model validation, and this had a significant, it has good prediction, and it uses 7 parameters like whether there are overbleeding manifestations, whether there is evidence of capillary leak, is there a hypotension or oliguria or anemia, or a delay to ASV administration. So based on these parameters, it was able to predict the mortality very well. This has been published. The next in the snakebite arena is there was an international various research groups working on snakebite got together and they wanted to develop a global core outcome measurement set. So what are all the important outcomes which you have to measure in a snakebite trial? So it's a collaborative effort, and Dr. Chanavirpa was part of that. And generally one of the focus areas is COVID, infectious diseases. We had studies on COVID. Scrub is an important area for us, and tuberculosis, meningitis, Myeliidosis is coming of late. Of course, sepsis, bloodstream infections, VAP are the regular load and problem we face. Some of the research currently on TB meningitis, an important trial is being ongoing with funding, extramural funding. Dr. Adaravan is the PI. It compares the usual, we have RHE, Z plus RHE, the usual short course therapy. This attempts at rifampicin at a higher dose of 25 mg per kg instead of the usual 10 mg, plus substituting ethambutol with moxifloxacin during the first 2 months. But continuation phase will be with the rifampicin INH, and instead of ethambutol using pyrazinamide for follow-up. So some tweaking and intensification with a high dose rifampicin with the 3 arms. One is the conventional, other is the modified intensified ATT short course, and third is the addition of aspirin. In TBM, infarcts happen, so aspirin could help. So this is a 3-armed trial currently being done. Hopefully it will have an impact. Then a very interesting Colisep study where colchicine will help in sepsis. This was taken by Dr. Venkateswaran. You know, the colchicine has inhibitory effect on the neutrophils. So maybe it will prevent the hyperinflammation of sepsis is the hypothesis. And this was tried in animal studies, like all drug developed in the sepsis. It worked very well in the animal study. It reduced the acute lung injury. It reduced the pro-inflammatory marker levels like TNF-alpha and STREM1. It lowered the pro-inflammatory, elevated the anti-inflammatory like IL-10. It had a good effect in animal model, but of course, human studies, we used a small dose because this is the first time colchicine is being used in a human study in the whole world. No one has used in sepsis, so the dose used was small. Of course, it did not have any significant effect, though it lowered the pro-inflammatory marker. This is yet to be, final data is yet to be analyzed, but overall there is no clinical benefit. Of course, the dose used was very small, 0.5 mg OD. We are very conservative because colchicine has not been used in sepsis anywhere in the world. So we have to be very careful about the dosing. So this is what it is. Maybe it needs higher dosing, longer courses, some modification with the experience gained. But an interesting area. This is the what Dr. Venkateswaran got an award for the best faculty research in progress last year. And we looked at ESBL bacteremia, not a great study, but we looked at what we are doing. There was a trial called Merino, which suggested that maybe carbapenems are better for ESBL pathogen, but it had a lot of limitations. So some centers have switched to using meropenem upfront for ceftriaxone resistant gram-negative pathogens, which are ESBL pathogens. But we still stick to use of capaparazone sulbactam or piperacillin tazobactam. We looked at how are we performing. In ESBL, we only 6% received carbapenem. Mostly we have used BLBLI. And our mortality was 26% in hospital and 28 days was 32, which is similar to other trials of gram-negative sepsis. So I think even though we are avoiding carbapenem, very limited use of carbapenem, but still our mortalities are not that much greater. So it's kind of reassuring in the interim. The Merino trial had a lot of limitations. It's being kind of similar trial is being replicated. Hopefully the results will come and guide us. But in the meantime, we are sure that we are not too much off the mark. Lata, how much time I have left? Lata? No, 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 no. Some idea should be there clearly. So, currently, I am timeless. Can you tell me where? No, no, how much time? Just okay, fine. We have also found an interesting link between OP poisoning as the cause of need for ventilation and staphylococcal pathogen. So it needs further evaluation. 15% had a staphylococcal VAP and 85% had a non-staphylococcal. OP poisoning as an indication was an independent predictor of staph. So we are looking at in a retrospective study. Prospective study is being undertaken currently. And of course, in our department, Dr. Hamid was daring enough to take a PhD candidate also, even though he was close to his retirement. In the last 5 years, he has guided a PhD candidate. We should all learn from him. Even at that age, he was willing to take up a responsibility of guiding a PhD candidate. And this was a study on polymorphism, genetic polymorphisms in nuclear factor kappa, beta, and TRAF3 genes, and how it affects the susceptibility to myeloma as well as how it affects response to bortezomib-based therapy. They did the SNP analysis and they found that a certain TRAF3 allele was lower in those with myeloma. Maybe it was protective against myeloma. A certain alleles of TRAF3 and NF-κ beta, they predicted poor response to therapy, and certain alleles indicated higher, better progression-free survival. Since today seems to be an oncology day, we also have an oncology data. The supplement. And so this is some of the research which some of it is impactful immediately, some of it is useful. I am not listed. This is the top 10 publication based on impact factor. We had an interest trial on scrub which got published in NEGM. Dr. Mukta, she will present that data. And we had other good publications in high impact journals with good citations. Some of it I've told, the global outcome, varus plotting, and the vaccine trials, and the infrared imaging. So some of it I've alluded, that's what I presented some of the data. So this is the publication citations. And of course, the number of research publications, we have about 40 publications per year. I'm not put them. Last 5 years, around 200 publications. Our funded projects, about 8 projects are extramural funded and about another 10 projects intramural are ongoing. The total funding for the last 5 years is about 4 crore. About 3.5 crore is extramural. About 50 lakhs is JIPMER funding. I have not put an exhaustive detail. Of course, there is awards for, I have not listed also for want of brevity. Next, I would like to ask Dr. Mukta to present the Scrub-typhus trial, which assess the role of doxycycline plus azithro versus monotherapy. And there is also further additional trial in Scrub-typhus. She will tell me. Thank you for the time and the opportunity. And once again, I express the thanks for the Dean Research and his team for, it's quite a onerous task of organizing and conducting. Thank you very much. Thank you.
I'll be presenting a few slides about interest trial. This was a randomized control trial, a multicentric trial, which was led by CMC Vellore. We were part of it and we contributed around 141 patients. That was the second largest compared to the after CMC Vellore. So as we know, Scrub-typhus is caused by Orientia sugamashi, and the vector is the larva of trombiculid mite. And the mite bites the human, penetrates the skin, and leaves an eschar behind it. That has a cigarette burn kind of appearance. Now, this Orientia goes and inhabits into the endothelial cells and can have multi-system involvement. So this study was for those who had severe scrub typhus, meaning that they were proven to have scrub typhus by RDT, a rapid diagnostic test, or an SCAR was present with fever, and they had at least one organ involvement, either a cardiovascular, respiratory, liver involvement, or CNS involvement, or renal involvement. So this was a 3-arm study, and around 1,000 patients were screened, of which around 809 were enrolled, and they were divided into 3 arms. The intervention was a combination of intravenous doxycycline plus azithromycin, and it was compared against the monotherapy arms. As we can see in this graph, the primary outcome was a combination of 3 factors, either a mortality at 28 days, or persistence of fever at 7 days, persistence of complications at 7 days, and fever at 5 days. So the outcome was compared as a difference in the risk, and if the patient was on combination therapy, the outcome was lesser when compared to the monotherapy groups, and the risk was calculated as risk difference. So there was nearly a difference of minus 14 percentage points in those on combination group. They were less likely to develop the primary outcome of mortality or persistent complications. On comparing the side effects, there were no differences. Those who on doxy plus azithro did not have any extra side effects when compared to monotherapy. This again shows the risk difference. It's it is better to be on doxy and azithro for a patient who has scrub typhus. And this is the analysis which shows that at day 7, the graphs have separated. That is probably why the patient did better on combination therapy was because the likelihood of having persistent complications on day 7 was lesser if they were on combination therapy. They were less likely to have persistent complications on day 7. That drove the outcome in favor of a combination therapy. Also, buffy coat of the patient's blood was examined for the persistence of the bacterial DNA by PCR, and it was found that in those who were on combination therapy, the bacterial eradication happened earlier than that in the monotherapy group. By day 7, almost the bacteria had been cleared in those who were on combination therapy. Now, this is very important because in a patient who is sick because of organ involvement, longer stay means longer ICU stay, longer risk of other complications, and in a resource-limited setting, it is difficult economically also. Now, whether these results were comparable or generalizable to our population, when we compared the interest trial participants' profile with those on published data sets and those what's available in literature on Indian general population, it was found that most of the characters were similar. That is, interest trial participants were mostly male, a slightly higher age, and the most common comorbidity was diabetes. So to conclude, the combination therapy was found to be more effective in those with severe scrub typhus. That is, they have scrub typhus with one organ involvement when compared to monotherapy with either IV doxy or IV azithro alone. Thank you.
Thank you very much, Dr. Mukta, and congratulations once again to have a publication in NEGM. And we're happy to see the progress of the department. I'd like to thank Dr. Vivekanand for giving an overview of the department. Thank you. Thank you very much. With this, we'll conclude the session of the departmental presentations.
Thank you, sir and ma'am, for navigating that engaging session. We have now reached the final and most anticipated segment of today, a testimony to the hard work and perseverance of some of the most talented and innovative scientists we have seen. It is at last time for the prize distribution ceremony. Although not everyone will walk away with a prize today, we are all winners who in our own ways contribute to the vast ocean of knowledge. For the prize distribution ceremony, I would like to invite on stage the officiating director, Dr. Bhavna Bade, the Dean Academic, Dr. Vikram Kate, Dean Research, Dr. D.M. Thapa, Student Advisor, Dr. Lata Chaturvedra, Convener of the Scientific Committee, Dr. Sonali Sarkar. Over to Dr. Nisha, the Associate Dean Research, to announce the winners.
Today we'll be distributing prizes for the first and second prize winners in different presentation categories. The presentations were held over the last couple of months. We also have 22 awards for GJ straws. They will also be given away today. So in the presentation categories, we'll start with the undergraduate presentations. The first prize goes in that category goes to Abhinav Sai Krishna Madipati for his work on the prevalence of gustatory function abnormalities among cancer patients receiving chemotherapy. You heard him talk some time ago. Co-authors were Suhas Ramamurthy and Prashant Ganesan. Second prize, Akula Bhavana Srinidhi for her work on perceived prioritization challenges and barriers to HPV vaccination by medical stakeholders, a mixed method study. Co-investigators include Ismail Rifai, Mahalakshmi, and Arpita. In the postgraduate category, the first prize goes to Jankala Anusha from PSM for this study of indoor air quality of houses near roadways in Pondicherry, and you heard her speak some time ago. Dr. Palnivel and others from the Department of PSM were investigators. The second prize goes to Aisha from PSM for her study on the utilization of supplementary nutritional services of ICDS through Anganwadi centers with Dr. Jayalakshmi. Junior residents, the first prize goes to Palni Raja, radio diagnosis, for the study that he presented earlier today in comparison of ultrasound guided surgical clip and commercially available breast marker clip placement with Dr. Sunita, Dr. Gomati Shankar, Dr. Bhavna Bade, and Dr. Vishwajit Bash. Second prize goes to Dr. Arihan Jain from Emergency Medicine for his study on the prognostic utility of peripheral perfusion index for 24-hour all-cause mortality in hypertensive trauma patients presenting to the emergency department with Dr. N. Balamurugan and Dr. Harichandra Kumar. Among senior residents, the first prize goes to Dr. Mahadevan from PSM. Study on enhancing data analysis in medical students, a competency-based training program using the DOAP method with Dr. Karthik Balaji and Dr. Avinash Kumar. The second prize goes to Dr. Rajashekar from Anesthesiology and Critical Care for a study on the comparison of subanesthetic ketamine and lidocaine as an adjuvant propofol-based TIVA on propofol requirement in patients undergoing emergency decompressive craniectomy or TBI. It was a randomized control trial The other investigators were Dr. Srinivasan, Dr. Prasanna Bidkar, and Dr. Satya Prabhu. Research staff, the first prize goes to Kavya from Medical Oncology. The study was titled Chemotherapy with Low-Dose Immunotherapy as Salvage Regimen in Relapsed or Refractory Hodgkin's Lymphoma, a phase 2 study with many co-investigators from Medical Oncology. The second prize is also from Medical Oncology, Emily Immaculate Mary. And that study is titled Bridging the Gap in Cancer Follow-up, Evaluating Teleconsultation Acceptance Among Survivors. It's an interim analysis of that study. Multiple co-investigators from Medical Oncology and other departments. PhD first prize goes to Akshata from Medical Oncology for her study on the assessment of imaging biomarkers to predict pathological complete response in patients with non-metastatic triple-negative breast cancer using a window of opportunity design. Second prize goes to Dr. M. A. Ramesh, Medical Oncology. No. Sorry. Yeah. Medical Oncology. Spontaneous apoptotic index, early apoptosis, and BAX, BCI2 protein expression as prognostic markers of ALL. Faculty research in progress. The first prize goes to Manu Iyen from EMSB for a study on developing a high-quality patient-centric integrated model for emergency care system in selected districts of India and implementation research protocol. We have 2 second prizes in that category. One is Anusha and from Pharmacology. Her study is titled Development and Validation of Self-Administration Medication Error Tool in Patients of a Tertiary Care Hospital. And the other second prize winner is Dr. Shivamurugan from Pediatrics. He did a run. This was an RCT on the effect of heat and moisture exchanger versus heated humidifier on ventilator-related adverse events in children. The prize for the best published faculty paper goes to Dr. Mrinalini from the Department of Pharmacology for her paper on isoniazid monoresistant tuberculosis treatment outcomes in Pondicherry, a mixed method study. Now we have the G.J. Strauss Awards. So first, Abhinav Sarvesh, here. Auditing of medical certificates of cause of death, pre and post introduction of immortality software in medicine department, a retrospective cross-sectional study guided by Dr. Ramdas from medicine. How to Pallavi? Can you all come forwards, please? The G.J. Strauss awardees. If you sit here, it'll be easier to come on stage. Pallavi's project Pallavi's project was on the assessment of sleep quality and its influence on mental status and physical activity among medical undergraduates. with Dr. Basanta Manjari Naik from Physiology. Next, Bobby T. Manideep Reddy. Prevalence of rare blood groups in undergraduate medical students of JIPMER with Dr. Aparna Krishna from Transfusion Medicine. Chinua Pailu. Early onset culture-proven neonatal sepsis and maternal genital colonization, a retrospective cross-sectional study in JIPMER from Department of Neonatology with Dr. Murugesan. Divyasri Suresh Babu. Assessment of text neck, forward head posture, and its association with cardiac autonomic status and psychophysiological disabilities in young and middle-aged individuals, a cross-sectional analytical study with Dr. Rajalakshmi from Physiology. Shashank. GVSVS Shashank did a comparative analysis of snoring on glycemic and hypertensive control in individuals with diabetes and hypertension. This was a project from ENT with Dr. Shivraman. Lavanya. Lavanya's project was on survival outcome of patients with muscle invasive bladder carcinoma following radical cystectomy, a retrospective study from a tertiary care hospital in South India with Dr. Shrira KS, Urology. Next is Niranjana Pinayar. Her project was on the length of hospital stay of preterm infants, a retrospective cohort study. I was the guide. Om Paddaredipalli. Burden of diabetic retinopathy and the protective factors against its development at the tertiary care center with Dr. Suryanarayana from medicine. Sonit Sai Reddy. Sonit Sai Reddy Vasipalli's project was on the comparison of long-term outcomes of alteplase versus tenecteplase thrombolysis in patients with ischemic stroke, an ambispective follow-up study with Dr. Sunil Narayanan from neurology. Suhas Ramamurthy. Suhas's project was on the assessment of functional status and quality of life among the patients undergoing treatment for spinal tumors or metastasis in a tertiary care hospital with Dr. Deep Sharma from orthopedics. Surender. Surender's work was on the association between two-wheelers vehicle types and causes of death among road traffic accident victims, a 3-year retrospective study in South India with Dr. Ambika Prasad Patra from Forensic Medicine and Toxicology. Tarunashri Paidi. Tarunashri evaluated visceral fat indices as markers of cardiometabolic disease in psoriasis with Dr. Medha Rajappa from Biochemistry. Talakola Sai Kiran Reddy. He studied the frequency of drug resistance among persons with idiopathic generalized epilepsy in a tertiary care hospital in South India with Dr. Pradeep Pangajakshan Nair from neurology. Vineet Reddy. His project was on the transitioning to a general surgery residency from undergraduate training, a phenomenological study of first-year residence experiences with Dr. Kadambari from surgery. Surya Teja's project was on the development and validation of low-cost simulation tool for practical training in diagnostic tracheobronchoscopy for otolaryngology postgraduates. Guide was Dr. Lokesh Kumar, ENT. And we had projects from nursing and other sciences, allied sciences also. Agila's project was on the comparison of risk of eating disorder between exercises and non-exercises among nursing personnel at JIPMER Puducherry with Mrs. Wembu from Nursing College. Aishatul Amana. Mana's project was on the assessment of factors affecting activities of daily living and instrumental activities of daily living among elderly patients admitted in JIPMER Hospital. This was a project done with Mrs. Shanmuga Priya from the Nursing College. Rosalind. Rosalind Priyadashini's project was on the assessment of parental burden and coping strategies among parents of children undergoing treatment at RCC. This was a project with Mrs. Angeline Mary Sheila, Nursing College. We also have Shakila from BSc Nursing, who studied the prevalence and pattern of psychiatric comorbidities and its relationship to metabolic risk factors among hypothyroid patients attending endocrinology OPD in JIPMER with Dr. Janardhanan from Nursing College. Naina Payal, BSc AHS, for the project on redefining hematological stability by understanding the effect of priming fluid combinations in RBC volumetric parameters during cardiac surgery in children undergoing cardiopulmonary bypass with Dr. Ram Shankar, cardiothoracic and vascular surgery. Angel Anna Matthew, BMLS, for the identification of free-living amoeba in selected water systems of JIPMER Puducherry by using polymerase chain reaction with Dr. Nonika Rajkumari, microbiology. Thank you.
Congratulations once again to all the prize winners. Your excellence continues to set a benchmark that all of us hope to achieve. But all good things must come to an end, and we have reached the end of the 10th annual JIPMER Research Day celebration. It has been my honor and privilege to compare for this event. Before we part, I request Dr. Subita L., Associate Dean Research, to deliver the vote of thanks.
Good evening, everyone. With this, we are coming to the end of another successful edition of JIPMER Research Day celebration. At the outset, I would like to thank our Director, Madam, for gracing this occasion. It was very special for us on this Women's Day to have you here with us, ma'am, for the Research Day observations. Thank you very much. I'd like to thank Dr. Gautam Roy for having given his guidance and support in the initial planning of Research Day, and our Director, Dr. Veer Singh Negi, for his approvals and guidance for conduct of this event. Special thanks to Professor Durarajan sir, our MS, and our Dean sir, beloved Dean sir, for his guidance and help. Without the support of academic division, I don't think the visibility of research would have been this much covered for the undergraduate students. And their support is very essential for us for conducting undergraduate programs and sensitizing them towards research activities. I would like to place on record thanks for the Dean Karekal for giving wide coverage for this event at their campus. Again, thanks to our nursing college administrator, the principal, for continued support for conduct of research activities and participation in the research day. Special thanks to the heads of department who've been present here throughout the day, and for the special presentations from the 3 departments today. So I think it's a second cycle that we have started for the different divisions, and we've seen a huge improvement over the years in the department's infrastructure and the research outputs. It was an excellent presentation. Thanks to all the heads of departments and the faculty who presented their and showcase their research work. And all the special invitees and other speakers, again, all the activities like debate and made this event very memorable. Now going on to the organization of the event, the scientific committee is the heart of such an event. So it was a big committee, actually. It was reconstituted this year, including many of the new faculty who have joined. So we saw a lot of ownership from them in conduct of these events. So thanks to all of them, and special thanks for the Dr. Sonali Sarkar, Dr. Gomati Shankar, along with Dr. Nishad. So this time again, they've tried, they never tire of work or effort. So they tried another method again for giving due credit to the people who present. So again, thanks for the continued efforts. A special thanks to Professor Chitoria and the IT team. For without their presence, this event here and in the other campus wouldn't have been possible. So along with that, I would like to thank the other members of the Research Day Committee. For stage management, it's always Dr. La. If Madam is there, then we are very comfortable in organizing the events. So along that, we have other members of the committees as well. For finance, Shri Hava Singh and his team members. Prize and certificates, Dr. Ilamurgan, along with other newly inducted team members. So again, it was memorable to see different, actually we had a separate prize categories this time. So all of them were done seamlessly. Food and catering, Dr. Chitra and other team members. So it has been a month-long event, be it the workshops or the different presentations. And this, thanks very much, ma'am, for this. Actually, you conduct it as if it's your house function every time. I don't know how you do that. Thank you very much. And for the souvenir, Dr. Deepak Kamalnath, again, for flawlessly bringing it out. Here, I would like to again place and record the image cover of the souvenir. Again, this is the second year. I think it is designed by Dr. Nishad. So please take a moment. We've circulated that in the groups as well. Request the faculty to give to circulate it amongst your groups. We'll try share it with the students. So please have a look at the souvenir to see the activities of the research division and the institute faculty and students that have been showcased there. Again, to Dr. Yogesh and his team for the banner and the display, not only today, for the continued 1 month activities. Special thanks to Dr. Ramesh and the other auditorium team members. So it's like our home and we come here every time for the research day function. Regarding the capacity building workshops, again, as mentioned, special thanks to all the coordinators, Dr. Palnivel, Dr. Sitanshu, Dr. Mahalakshmi, and Dr. Chakradhar for the different workshops. Now, coming to our home department, so special thanks to all our staff of the Division of Research for all the support that they have rendered in all the administrative activities, approval and support, Mr. John Peter, our AO at the department, and also at the finance division for whatever research output you have seen throughout the day is not possible without a steady support in the management background. So all the staff, and special thanks to Mr. Yuvraj here, so for his continued presence and support. So we were able to more or less hand it over to them completely. So they're able to conduct it as if it's their department function. So thank all the JIPMER faculty, PhD scholars, students, and other project staff who participated in all the events and made this a wonderful, successful celebration. Finally, I'd like to thank our team members, Dr. Vikas in his absence. So he's done all the work and given us a wonderful chief guest. So in his absence, I place on record thanks to Dr. Vikas, Dr. Nishad, and Dr. Prashant Ganesan. So every time it's always to see how best we can improve one research day experience to the other. Then finally, it's thanks to Dr. Thapa. So sir, for your support. So wherever we miss, sir is there to guide us and support us. And he does that in a very, at that moment, it is very stern and strict. But at the end, we see a steady guidance, support, and love at the end. Thank you very much for your support. So if I've missed anyone, kindly excuse me. And thank you very much. A very memorable day. One more feather in the cap for Research Division and for JIPMER Institution, for all the faculty who made this a wonderful occasion. Thank you very much.
Would request everyone to please stand for the national anthem.